Thank you and welcome to this last session, close to my heart, on the bacterial fitness. I'm Bouke de Jong, I'm one of the chairs.
I'm Rajni Rani from National Institute of Immunology, basically an immunogeneticist and immunologist, working on infectious and autoimmune disorders.
And I'm Frank Koblenz, I'm an epidemiologist working at the University of Amsterdam in the Netherlands.
So it's my great pleasure to introduce Caroline Williams, a clinician from Leicester University. And her group, including Mike Barrer, should really get the credit for pioneering the RNA extraction from sputum. Caroline, please.
Okay. Good afternoon. Mycobacterium tuberculosis is an obligate pathogen. It requires transmission in order to survive. Therefore, it must alter and adapt in order to be aerosolized and in order to be ready to infect the onward airways. It's therefore unsurprising that it should, in some way, change in order to adapt to its new environments through the stresses of aerosolization and transmission. Unfortunately, accessing the samples that allow us to understand these changes and adaptations in the process of aerosolization and transmission have so far been limited. Our group have been looking at ways of trying to capture and understand the aerosol over the last few years, and I'm going to just take a few minutes of your time to explain some of the mask and aerosol sampling that we have done. Airborne transmission was first demonstrated by Wells, Riley and colleagues in the 1960s when they looked at skin test conversion in guinea pigs that had been exposed to room air from pulmonary TB patients. And in vitro experiments have really described the importance of these fine droplets within the respirable range in the transmission of TB. Clinical studies have also shown that the majority of particles and aerosols expelled by patients with TB are within that 1 to 5 micron respirable range. However, it's not all that they give out. To our left, you'll see here that these are a range of aerosols captured in, culturable aerosols captured by the Finelli group and also by Robin Wood's group in South Africa using a size cascade impactor, and you'll see while the majority of the aerosols do fall in the stage 4 and 5, the respirable range under 5 microns, there is a diversity in that range. And the role of these larger and potentially smaller aerosols and droplets in transmission is currently unknown. The traditional view of aerosol formation is that it's done by shear forces. Forceful respiratory maneuvers like coughing and sneezing shear particles and aerosols from the fluid within larger airways, and these are expelled as aerosols, with the rest of it being expelled as sputum. With this view, it would make sense that there is a relationship between sputum and aerosol, that aerosol is perhaps a subset of sputum. More recently, work of Gerhardt and colleagues has looked at the bronchial fluid burst mechanisms, which basically says that in the smaller airways, as we expire, the airways completely close, a film covers that, and then when inspiration starts, it snaps open, and aerosols are created from the burst of that fluid. This is done with tidal breathing. It doesn't necessitate cough. The variation in the quantity of bacilli excreted by patients and the infectivity of patients has been widely observed in both clinical studies and in clinical practice. But what we've never really understood is the pattern of spontaneous MTB excretion by individuals over time. So capturing aerosols isn't as easy as you'd hope. We've isolated aerosols indirectly from patients with pulmonary TB and using either the room air or highly specialized research chambers such as these two. So on our left, we have a respiratory aerosol sampling chamber from South African group where a patient sits within the chamber and the air that they exhale and within the chamber is captured and then either the aerosols are either cultured or undergo PCR. With the Finelli-designed cough aerosol sampling system here, a patient coughs into a drum, the air is pulled over a size differentiating cascade impactor. It's only the work of Eddie Jones-Lopez and Kevin Finelli using this cough aerosol sampling system that has looked at an individual's contribution to transmission. And in fact, they found that that culturable cough aerosols predict better than sputum AFB grade and individuals' infectivity in household contact studies. So we thought, is there a better way of capturing aerosols directly from patients? And I know that you've had a talk about the Mumbai group that have also used face masking sampling. But we started looking at it to see whether we could capture both replicating and non-replicating MTB directly from patients by wearing a face mask. One of our colleagues, Eddie Cheetah, started the ball rolling by using a bacteriophage, and he was able to isolate replicating MTB with 15 minutes of somebody wearing a surgical face mask. We then modified the design to include a gelatin filter, as you'll see in this picture, and tested it on patients with pulmonary TB in the UK and in the Gambia. We're able to isolate and detect both non-tuberculous mycobacteria and Mycobacterium tuberculosis with a sensitivity of about 65% and a specificity of 100%. Importantly, we were able to detect MTB from patients that required a bronchoscopy to make a diagnosis as they were unproductive of sputum. And as I've mentioned, subsequently, RNA has also been able to be detected from face mask sampling from pulmonary TB patients here in India. So I just want to talk to you about our first study using mask sampling. And the idea was to try and understand the relationship between what people exhaled, expectorated, and excreted over 24 hours. And to see if we could understand that relationship in the context of clinical markers of infectivity. So we found 24 hospitalized TB patients in 3 hospital sites in Pretoria in South Africa. They were already diagnosed with TB by either gene expert or AFB grade. They were pre-treatment or within the first 24 hours of treatment. You'll note that this cohort, because they were hospitalized, were sick. Their majority were HIV positive and heavily immunosuppressed with a mean D and CD4 count of 35. What we did was we asked patients to wear a face mask for 1 hour every 3 hours. So they wore it for an hour, then they had a couple of hours off, then they wore it again for another hour. We collected all the sputum over 24 hours, and when they were wearing the mask, it was observed. We asked them to wear a Leicester cough monitor, which is basically an MP3 player with a microphone that's clipped to them for 24 hours, and it counts the cough using semi-automated software. We then extracted the DNA and quantified it using IS6110 and a subset using RD9 probes. We used background controls as well. So, just want to show you, this is basically the patterns when we put together for aerosol, for sputum, and for cough. So, I appreciate the axes haven't quite displayed correctly, so let me look at this one with you. This is up to 10 to the 9 copies of IS6110, and this is up to 250 coughs frequency per hour. I'll draw your attention to the green line, which is the aerosol burden found in mask samples over that 24-hour period. 0 is midnight, and then you go up to 11 p.m. And then cough frequency this is the average number of coughs per hour over that 24-hour period with the blue line. The red triangles is sputum if it was expectorated spontaneously, and will also tell you the burden of IS6110. Here are 2 examples of the heterogeneity that we really saw in our patients. The first patient on the right had over 2,000 coughs in 24 hours and also produced sputum throughout those 24 hours, but wasn't always containing bacilli. The aerosol production by this patient was also highly variable, with spikes of up to 10 to the 8, but also having some samples that were negative. Then if you look at the other patient, patient 21 next to him, he only produced 1 sputum sample, which had nearly 10 to the 9 copies of IS6110 in, had a cough frequency of less than 50 over the 24 hours, and produced no bacilli at all in a mask sample. This is all 24 patients. I don't want you to necessarily critique every single graph on there, but to really appreciate the groups that we think we've observed here. 16 of the 24 patients had a pretty consistent aerosol output. So remember, aerosol is green, cough is blue, sputum is red. So they continually produced aerosol at a pretty flat linear rate over the 24 hours with a range between 10 to the 3 and 10 to the 5 bacilli in each 1 hour sample. We then had 6 patients that had a more variable pattern of aerosol production along the bottom here, 2 of which had high peaks, again around 10 to the 8, 10 to the 9, these two down here, but 4 had much lower peaks. And then we had 2 patients that produced absolutely no IS6110 in their mask outputs over the 24 hours. The numbers are small, but the frequencies are pretty consistent with the high variable aerosol producers that Rod Escombe found in the Lima study of HIV-positive patients, and provides synergy with the super spreader phenomenon potentially that we're seeing. So small people that produce large amounts of aerosol. But we were noticing that it was in pockets and spikes rather than a continuously high output. This is another way of looking at the data, which is just to really drum home the heterogeneity, but this is the full 24-hour totals. So again, green is aerosol, red is sputum, blue is cough. And you'll see that there are some patients that have absolutely loads of IS6110 in their aerosol, but are unproductive of sputum. So their red dots are at the bottom of the axis. Then conversely, there's people that produce no IS6110 in their aerosol, but produce up to 10 to the 9 copies in their sputum over 24 hours. And I hope you can appreciate on this graph that there doesn't seem to be a relationship, or in fact it seems random, between cough and what they're producing both in sputum and in aerosol. So we thought we'd look at that a bit more. The median cough frequency for this group over 24 hours was 466 coughs, which is very high. On average, we cough 5 times an hour, so this is much higher than that, which is surprising when you consider that this is a HIV-positive dominated cohort. We found no association between MTB output in aerosol and corresponding cough frequency when we analysed it hour by hour over the total 24 hours and over the total 8 mask samples taken in that 24 hours. What was more striking was when you looked at when the patients were sleeping. So remember, these guys were observed, so we knew when they were sleeping. And we looked at the coughs that people coughed when they were asleep, and you'd be surprised to know that we don't all cough in our sleep. And you'll still see that they produced aerosol IS6110 when there was no coughs in that hour. And that was from 6 participants. So we looked at those 6 participants, and the amounts of IS6110 captured when there was no coughs was in keeping with the amount that was captured by the same patients when they were awake and coughing. It's also keen to note that there is quite a lot of IS6110 captured when there's less than 5 coughs an hour, so less than the average healthy person. Just to really sort of highlight this disparity between cough, if we then look at the converse, if we look at times when no MTB was captured in mask outputs, people were still coughing quite a lot. So up to 318 coughs an hour and absolutely no bacilli excreted onto a mask. Finally, we thought we'd look at the traditional markers of infection that as clinicians we use to make public health decisions about whether this person is highly infectious or not, and whether that correlated in any way with, if that correlated in any way in mask output. The presence of cavities and the extent of disease on chest X-ray had no correlation with mask output. The bacterial burden from the baseline sputum taken prior to starting the study also had no correlation, both looking at the burden by AFB grade, expert, and time to positivity in midget. When we looked within the 24 hours studied, there was again no correlation between what we caught in masks and what we caught in aerosol. So whilst this study initially was not designed to look at diagnostics, it struck us that within each 3-hour period, we were more likely to get a mask sample. In fact, we got one every single time, whereas we only got a sputum sample around a quarter, 25% of the time. We were also more likely to detect MTB in a mask sample at 166 out of 192 times, whereas we only managed to get it about 20% of the time with sputum. So it caught our fancy. Maybe mask sampling could be used as a screening and diagnostic tool. So we did a small pilot study in Pretoria in South Africa by joining a TB outreach screening programme at an informal settlement in the city centre. We screened 48 people using the WHO screening criteria, and 20 of which were symptomatic and had 1 sputum and 1 mask sample taken. They only wore the mask this time for 30 minutes, and we used a slightly different membrane. Both were analysed using the GeneXpert platform. So 12 out of the 20 patients were both mask and sputum negative. That's leaving these 8 here on the right. As you'll see, 1 of them was positive by sputum only, 1 of them was positive in both mask and sputum, and then that left 6, all of whom were positive by mask. This was not what we were expecting. And to be honest, we hadn't planned on following these guys up with any more investigations. The idea was to see, as a diagnostic screening tool at one time point, could we predict whether they had TB? They were a transient group of people in a squatter camp, so chasing them down was actually quite hard work. And by the time we got hold of them, 1 had been lost to follow-up because he'd moved to Cape Town, and the 2 with positive sputums had rightly been started on TB treatment. So then if we look at these 8 patients, by the time we followed them up at 6 weeks, 5 of them were sputum positive, 4 of them were sputum positive, by taking multiple sputum samples, and 1 of them was negative. All of them had normal chest x-rays, so we undertook PET-CT scans. The PET-CT scan showed early lung disease in the 4 patients that were also sputum positive. So they were commenced on treatment, and at follow-up halfway through treatment, both their sputum, mask and PET scans were now negative. That left us with this 1 patient at the top. The mask was still positive, but the sputum was negative, and on questioning, she had recently been treated for TB, so we undertook one of her masks to have with propidium monoioside to remove any intact or free DNA. At this point, the mask became negative, and an alternative diagnosis was made. That sums up the quantitative part of our aerosol work, and I'm going to move on, if time allows, to talk about the quality of aerosol in aerosol sputuming. So we did a small pilot in 2015 from 5 patients using a modified cough aerosol sampling system where we asked patients to cough into RNA stabilizing liquid, and then we looked at the transcriptomes and microbiome captured. We noticed there was a substantial difference between the aerosol and the sputum microbiome. So we decided to follow this up. We looked at 22 naive sputum-naive patients, and 17 of them were HIV positive, and 20 had MDR or XDR. We collected aerosol samples in 2 5-minute coughing sessions into this GTC, and collected sputum at the same time. We then used the RNA sequencing to look at the transcriptome, compared with exponential H37RV. Basically, we found a very distinct difference between the aerosol and sputum transcriptome, and this can be highlighted here using the DOS-R regulon. Finally, we looked at the microbiome of these patients. The great thing about RNA-seq was we could look at all of the bacteria, and that included, and that allowed us to get a better taxonomy, or have more confidence in our taxonomy assignment, rather than just basing it on 16S DNA. This is the composite microbiome for sputum and aerosol, and I hope you can see that we think it is distinct. Finally, I just want to show you 3 examples of the heterogeneity we saw within the microbiome that complements what we were finding in our mask sampling. The aerosol microbiomes are at the top here, and the sputum is at the bottom. Patient 8 has mycobacteria in their aerosol microbiome, but not in their sputum. Patient 22 has it in their sputum, but not in their aerosol. And patient 13 has it in both. The striking feature of the sputum microbiome is the heavy dominance of Streptococcus and other upper airway bacteria, which you would probably expect in the formation of sputum. So I'll just finish by saying, hopefully we're opening a door. The talks from this afternoon will continue this to really be able to understand what happens to the bacilli during its flight from inside one person's lungs to inside the next person's. Face mask sampling may offer a non-invasive, clinically compatible tool to help us understand that and potentially help us make diagnoses. What I hope that you've been able to appreciate is that there are distinct patterns that are seen over 24 hours, but none of them seem to be completely the same between patients. Cough appears to be less important in aerosol formation and therefore in excretion and transmission. And the mycobacterium and its accompanying microbiome is very distinct in aerosol compared with sputum. I'd like to say great thanks to all the study participants, but to everybody's hard work in Leicester, in the UK and the US to make what I've just told you hopefully come to life a little bit. And with that, I thank you.
Thank you very much, Caroline. Given the time, we'll accept one question. Okay.
Hi, that was so interesting. I was wondering if you have plans or hypotheses about whether the Mycobacterium tuberculosis populations are different from the mask sample and the sputum.
I think they definitely are. I think the 24-hour mask, the mask work has definitely shown that the populations in quantity are very different. They don't ever correlate, and hopefully you've appreciated that the transcriptome is different, as is the microbiome. So I personally feel that the population of bacilli in aerosol is distinct of that in sputum, so would support the hypothesis that actually they aren't from the same source. So maybe they're not from the shearing forces and the cough necessary to produce the aerosol, and it's more likely that they've come by a different mechanism, and therefore that's why they're so very different.
Thank you very much.
Thank you.
May I now invite Dr. Edward Nardal, who is a professor of medicine at Harvard Medical School, associate physician at Brigham and Women's Hospital, Division of Global Health Equity and Pulmonary Medicine, professor of environmental health and immunology and infectious diseases at T.H. Chan School of Medical Health Sciences. Dr. Nardal's research interests focus on tuberculosis control in general and transmission control in particular. Dr. Nardal, please.
Thank you, and thanks for the organizers for this chance to speak at this very impressive meeting. So I realize that what I'm going to be saying about fitness is not as we traditionally think of microbial fitness, which has more to do with the replication of the organism and often talked about more in molecular genetics than at this level. But still, I think you'll see that there is some commonality here. I like this diagram as a way to put together the elements of transmission, beginning with the infectious source and moving through the environment to the host, and then back again to through organism progression to disease. And this particular version is designed to emphasize where interventions can take place. And the one that I've highlighted is prompt effective therapy. And I will make the case that there's nothing you can do to stop transmission better than to identify the patient early and start effective therapy. But only effective therapy. I mean, you can put a mask on the patient and that will be somewhat effective, and we can kill the organisms in the air and that has its effect. And we can vaccinate the host, and that's going to receive the organism, and that will have an effect, but nothing more important than treating. Now, in this version of the same diagram, sort of looking at the various environments that the organism is going from, certainly inside of the source, they can be inside cells, inside cavities, inside airways, and as Carolyn pointed out, there may be differences between organisms in sputum and organisms in aerosol. Then, once released, these organisms must adapt to a completely different environment of traveling through the air, where they encounter temperature differences, humidity, dehydration, rapid dehydration, and then enter into another host where they get rapidly rehydrated and land into a host where they are charged with overcoming both structural and immune host responses to cause infection. So a wide variety of environments. So we talk about fitness to infect. There are the innate fitness characteristics which Sebastian and others have described, where, for example, the Beijing strains and certain other strains seem to have greater ability to infect and cause disease, largely determined by mutations, presumably, and compensatory mutations, as we've learned about as well. In terms of the environment, there's a potential for loss at every single stage along the way, with responses to these environments. Mike Baer has described organisms in the airways as fat and lazy, presumably metabolically somewhat inert and carrying a lot of neutral fat. So again, an adaptation that we don't fully understand. And then in the new host, again, rehydration, et cetera. And then in terms of treatment, certainly one of the most important impacts on stopping transmission would be cell death. But it's likely, from what I'll show you in a minute, that there are sublethal stresses that may impact the ability to transmit, and that happens very quickly. Now, way back in '76, Annick Rion and others described the 2-week rule, suggesting that patients on effective therapy would be infectious for about 2 weeks, and then after that, perhaps not. And that was based on epidemiologic studies, looking at household contacts, et cetera, and was more or less a consensus. There was not much real science after that, except for the fact that some more household contacts were not infected after a certain period of time. Then, before that, actually, Richard Riley and the experiments that Carolyn alluded to set up an experiment in Baltimore where there were 6 hospital beds, and above the special ward, in a very tightly sealed ventilation system, were housed guinea pigs, hundreds of them, which would receive the air from the patient ward, and which you could use then to quantify the numbers of organisms, infectious organisms in the air by skin testing. And that work is all published and republished, actually, in a more recent journal of epidemiology where, 1995, it was considered a classic in epidemiology. In the second of his 2-year studies, Riley compared directly patients on therapy and patients who were not treated, where treatment was delayed. We might have trouble doing that today, but he was able to do that at the time. And both drug susceptible and drug resistant, and at the time the only drugs were isoniazid, streptomycin, and PAS. So not the most potent regimen in the world, but nonetheless, if the 61 patients who were untreated with drug susceptible TB infected 29 guinea pigs on the top line there, we have a pointer, okay. 100 is 100%, then taking into consideration the amount of time that the patients and the guinea pigs were exposed, then 29 treated patients infected only 1 guinea pig, so they were 98% less infectious. Of the drug-resistant TB patients, there were many fewer to compare, and Riley didn't really comment too much on these numbers, that the untreated drug-resistant patients seemed to be less infectious, and the treatment, although it reduced transmission, the numbers again were small. But these numbers are not as small, and I think are quite believable and have been reproduced, as you'll see. So what I missed about this experiment in the many times that I read it was that this effect was not after 2 weeks. It wasn't even after 2 days. The patients were admitted to the ward and started on therapy. So there was no amount of time of treatment that caused this effect. This was more or less immediate. Now, it takes some time to infect a guinea pig, so there was some treatment effect that was being watched here. But this isn't after 2 weeks of therapy. This is starting treatment at the same time. And in his own words, what he said was that patients were admitted to the ward and the treatment was initiated. And they were generally removed before the sputum became smear and culture negative. So sometimes casual observers think it's odd that you could be sputum positive on culture and not infectious. But there are many, many differences between those two measures. In sputum, we're doing everything we can to grow it. We put a large bolus of sputum into a culture medium. And in aerosol, we're doing a lot of things to kill off the organism, so to speak, aerosolization, and the host, on the other hand, is doing everything not to be infected as well. And so this discordance shouldn't be as surprising as it seems. Now, in terms of why this happens, which is the point of this talk, this paper from 50 years ago was by Loudon, and he postulated that if you had drug in your secretions, that as particles fall to the ground and evaporate, that drug necessarily must concentrate, and that this very high concentration of drug may account for the rapid effect of drugs on treatment. There was really not much experimental evidence for that, and I would say 50 years later we know exactly not one thing more about the effect of treatment on transmission than Loudon speculated on. We have reproduced the Riley facility in Pretoria, South Africa. It's called the Air Facility, and have been since 2005 doing transmission experiments from humans to guinea pigs. And over a series of several experiments, we looked at the transmission from 109 drug-resistant T patients who were admitted because they were smear-positive, cavitary coughing, and recently started on therapy. Could be days, could be or less than that. And these are some of their x-rays. Many of them were HIV positive, but still had cavities in many cases and lots of lung infiltrates. One of the things that was striking is that there was quite a bit of variation on the experiments. In our very first experiment, we infected over 4 months, 26 patients over 4 months, infected 74% of 360 guinea pigs. In subsequent smaller experiments, 24 patients over 3 months infected only 10%, 15 patients over 2 months, 53% of 90 guinea pigs, and in this experiment that I highlighted, 27 patients with all these same characteristics over 3 months infected one guinea pig, 1% of 90 guinea pigs. And then finally, in this last experiment, with no interventions, 17 patients over 3 months infected almost 80% of the guinea pigs. So as Carolyn mentioned, variability in transmission is certainly a characteristic, but this seemed extreme, particularly this one experiment. Until we got back some of the results of the few isolates we got out of guinea pigs, it's very hard for us to keep highly drug-resistant infected guinea pigs for any length of time for them to develop disease, but we did get some disease. And in many of these experiments, we were able to find XDR-TB before XDR-TB was actually described, but subsequently out of molecular testing, we found XDR-TB, except in this experiment. where there was no XDR-TB, where standard MDR-TB treatment completely suppressed transmission, just as the treatment with INH streptomycin and PAS had done 50 years earlier. So we've begun to do shorter experiments, and I don't know if this is more confusing or not, but basically we take, in this case, 5 patients, and we started them on the added bedaquiline and linezolid to their failed regimen, as is being done in South Africa. We studied them for 8 days before starting the bedaquiline linezolid, and in that 8 days, in one guinea pig colony, 24 of 90 guinea pigs were infected by that cohort. We then started treatment and waited 72 hours to have some opportunity for the drugs to take effect, and with the same patients, we studied their transmission to a second colony of 90 guinea pigs, different guinea pigs, same type, and got 25 of 90 infected. So essentially the same rate of infection before and after starting bedaquiline and linezolid for a total of 11 days. The 72 hours of treatment and then 8 more days of treatment. So no effect, which was surprising because up till this time we thought that effective treatment stopped transmission, and these drugs were added for greater efficacy. So we've been doing this again. We published, let's see, I'm sorry. This is the same experiment shown somewhat differently, a pre-treatment infectiousness of the same before and after starting bedaquiline and linezolid, 5 patients. We've done it again with 9 patients, and in this case we used the NICS regimen, which has been notable for its ability to convert sputum, et cetera. And we had a particularly infectious cohort to start with before starting treatment. Same idea, 72 hours of treatment and then second guinea pig colony exposed. And in this case, there was complete suppression of transmission with the addition of prothrombamide. So we were quite impressed that, first of all, the model can tell you whether the drugs are effective, and it can also tell you when they're not effective, which I think is very important. And it was reasonably efficient. This is a total of 11 days of treatment and another 8 days. So with, excuse me, in less than 3 weeks, we're able to assess the infectiousness of treatment on a cohort of patients. So what good is this information? In terms of how patients are treated, it's always embarrassing when you're talking about transmission and you can't stop coughing. I assure you this is a cold and not anything more. At any rate, we published the rapid effective treatment on transmission of multi-drug tuberculosis, assuring that ambulatory treatment of MDR-TB actually is justifiable based on the rapid effective treatment. And that actually led to an approach to infection control that says if treatment is so effective in stopping transmission, then most transmission must be coming from patients who are either not being treated or being incorrectly treated. And so when you look at, there are many studies of unsuspected, untreated patients who enter the hospital, not on the TB ward, but everywhere else, or on patients who enter the hospital are treated as drug susceptible, turn out to be drug resistant, and are infectious for perhaps months until they fail therapy and ultimately are diagnosed as drug resistant. Sorry. So, thank you. So this we call FAST, find cases actively, active case finding in the hospital, separate and treat effectively. So that's one use. But exactly how this stops, again, is not clear. An updated hypothesis, I would say, is that MTB has essential adaptations, as Carolyn pointed out, to airborne transmission and infecting a new host. And effective antibiotics probably impact these adaptive responses before killing occurs. Some effects that we don't know. So the question is, how could we study this? And I began to look at what's known about the transcriptomic changes of treatment, and there's quite a bit known, and it isn't simple. There's obviously, and this is a review of the recent review of the topic of what happens here. And the problem is that it's very difficult to separate out the responses of the stress responses and from and combinations of drugs from the effects of transmission on the transcriptome. So this paper concludes with the fact that there is progress in this area, adding metabolic data, proteomic data, and lipidomic data should bring new insights into what molecular networks are involved in response to antibiotic treatment. But they are complex, and we're talking about many different kinds of antibiotics, many of which seem to stop transmission. Now, you don't need transcriptomics in some cases. We've been done some experiments with colistin, a detergent-like drug that simply disrupts the cell wall. And here's some photomicrographs of organisms, scanning micrographs of organisms before and after exposure to colistin. And you can understand why that might impact transmission. These are grossly damaged organisms, probably not at the metabolic level but as much as structurally. And then finally, the adaptation of cells to new environments is a big topic. And we're talking about organisms that evolve from one animal to another over time, mutations, et cetera, but also adaptations to very rapid changes like going from sputum to aerosol and into the air and being dehydrated, et cetera. And this is perhaps even more complicated than the drug effects. And so I think the problem we face is that we could capture aerosols, as Carolyn is doing, and try to look for commonalities among drugs that do very well stop transmission and others that do it less well, like bedaquiline and linezolid, although that may be a drug concentration effect because they're slow to accumulate in the body. But teasing out the responses, the unknown, what we don't know about responses to transmission from what we don't know about responses to drugs is going to be very, very challenging indeed. So again, we are continuing to screen drugs and regimens for their ability to stop transmission. And one benefit might be they might show some potential targets that, although we would not probably develop drugs just to stop transmission, they may be informative in terms of new targets we haven't thought about because they're not really built around killing necessarily. Another approach might be to look at TB in macrophages before and after treatment, and that is actually being done, and look for common patterns among effective drugs. So I think we've known that there's a very rapid effect of effective treatment on TB transmission for the last 50 years, and that it happens well before sputum conversion, but the mechanism isn't known. And apart from killing, effective antibiotics may rapidly impact critical TB adaptations to the stress of aerosolization and airborne transport and or the stress of host defenses. The database of antibiotic effects is growing, but there's a lack of data so far on microbial adaptations and what's going on there. Is it, in fact, even all transcriptomal, or are there other protein-protein interactions or whatever that allow for very rapid adaptation to transmission? Stop there.
We have time for one quick question, please.
So, Ed, in the Riley Wells type facilities, the bacteria travel maybe 20 meters, something like that. Do you think that's enough time for the drugs to take effect, and maybe if you were looking at a closer point, they might still be infective? So, yeah.
Hard to know. Probably there is some time course. I think one of the other things we failed to appreciate is the probability issue here, whereas we put stuff in culture, it has all the time in the world to grow and et cetera. And here we're talking about a chance of being infected by a host and at a very, very low dose. So the, whether there would be whether organisms closer to the source would be infectious or not, I don't think we know. The probability of being infected would probably be greater, just by probability alone.
Thank you, Dr. Nardal, for that very interesting presentation.
Well, Sebastian is already there. It's my pleasure to introduce to you, probably known to many of you, Sebastian Gagneux, who is Associate Professor at the Swiss Tropical Medicine and Public Health Institute in Basel, Switzerland. He is a evolutionary biologist, infection biology, studying the ecology and evolution of Mycobacterium tuberculosis, with particular focus on determinants of host preference and drug resistance, and combining large-scale population genomic and genomic epidemiological studies in various countries. Sebastian, the floor is yours.
Thank you very much, Frank. And thank you very much. And thanks for the opportunity to come and talk about compensatory evolution in the context of transmission of multi-drug resistant tuberculosis. We heard about fitness of flying and fitness of infecting, and I might have used another complementary title, so the fitness of infecting and then causing secondary cases to really complete the life cycle of Mycobacterium tuberculosis, particularly in the context of multi-drug resistance. So here are my disclosures, and here are the numbers that we all know, the latest WHO numbers. There's one particular number I would like to highlight, which is this one here. So only 3.4% of new cases of TB are multi-drug resistant. Now I say only because this comes as a surprise if you think that TB has been treated for at least 50 years, and this low percentage globally has been rather stable over the last 10 years or so. So this would suggest that somehow multi-drug resistant strains are less transmissible, less virulent, etc. And clearly the picture is more complicated, because if you go into more details, we see that there are areas of the world where the problem is actually much more significant. So these brown areas, the former Soviet republics, we know that in some of these countries, primary MDR is up to 30 or even 40%. We know that there's many non-biological factors that likely have contributed to this problem. My group and our collaborators, we have been interested in trying to explore the bacterial factors that might have contributed or are contributing to the problem. The traditional view of looking at how drug resistance evolves in the context of fitness is really this idea that if a bacterium starts drug susceptible, it will often, but not always, pay a cost in reduced fitness, meaning reduced virulence, reduced growth rate, reduced transmission in absence of the drug. Now, because evolution continues, the next part of the scenario essentially is that you have compensation. So Ed already referred to these compensatory mutations, and this is a kind of a general phenomenon which has been observed in many bacteria, but also in viruses. And in TB, several years ago, we and others have discovered some of these so-called compensatory mutations in the RNA polymerase of Mycobacterium tuberculosis in various subunits. As you know, the main target of rifampicin is in beta subunit, so the red part of the enzyme, but these secondary compensatory mutations were actually found in various subunits, including RPOC, RPOA, and also lately in RPOB itself. Later, many strains, MDR strains, etc., have been sequenced by many groups, and these compensatory mutations keep popping up, so we see them a lot. We don't really yet understand what they do in the clinic, in epidemiological setting. We kind of know we think we know what they're doing in the lab, so they're clearly improving the fitness of clinical strains of TB when you grow them, for instance, in the laboratory. By the way, similar mutations have been found in other rifampicin-resistant bacteria. Other species are really supporting this idea that these compensatory mutations are actually relevant for the biology of the organism. The question is, how relevant are these for the epidemiology of multi-drug resistance? And that's essentially the question that we're asking, but also what might be the ecological drivers of these compensatory mutations. And so to address these questions, we started, I think it was early 2014, a nationwide study in the Republic of Georgia, a former Soviet republic, so part of these high MDR burden countries. Together with our collaborators of the National Center for Tuberculosis and Lung Diseases in Tbilisi. And then I'm essentially going to present data that Sebastian Gigli, a PhD student, and also Chloe Loiseau have been working on. So there's various data sets that we see. We started in 2014 with a prospective study. At the same time, we did a retrospective study, and that's the first part of the data I'm going to show. I presented this briefly last year, so I promised if I would be invited again, I could keep the more complete story in terms of what happens as far as the prospective part of this study is concerned. So let's start with the retrospective collection. So these were essentially all MDR plus, meaning MDR, pre-XDR, and XDR strains that we could resuscitate, if you will, from this collection. So it's about 659 strains across these 3 years, 2011, 12, and 13, which corresponds to a bit more than half of all the culture-confirmed reported MDR cases in the country. So this is the tree that results from these genomes, and maybe not surprisingly, we kind of expected that. There's an overrepresentation of lineage 2, which includes the infamous Beijing strains. 77% of all of these strains carried the compensatory mutations in any of these subunits I was referring to, which is actually a high proportion. And an estimated 75% of these mutations, these strains were clustered, presumably under estimate, given that we only sampled roughly half of all the MDR cases during the period. Nevertheless, we went ahead and tried to identify risk factors for clustering, which is a proxy for ongoing transmission of multi-drug resistance in Georgia. And so these are the results of multivariable analyses adjusted for age, sex, and previous TB. One factor was just the number of drug-resistant mutations, not a very strong effect. Lineage 2 popped up as quite a strong risk factor, so MDR strains that are lineage 2 are more likely to transmit. This was kind of already known. Then what we hypothesized originally, yes, so compensatory mutations do contribute to transmission independently of these other risk factors. And actually the largest variable which popped up was essentially a history of incarceration during the study period. Now, retrospectively, this was not necessarily surprising. We know that prisons play or have played a very important role, particularly in these areas of the world, as far as TB and also MDR TB is concerned. We hypothesized that perhaps incarcerating people might be also a way to somehow manipulate the evolution of multi-drug resistant TB strains. And the way we tried to explore this was essentially by reconstructing the individual transmission events, essentially trying to measure who gives TB to whom. So does TB travel from essentially a prisoner or ex-prisoner to a civilian or the other way around? And then see the role of compensatory mutations in these transmission events. And so if you do this and look at the instances of transmission events from a prisoner to another prisoner, we see that 91% of these transmission events essentially are associated with a strain or an isolate which carry a compensatory mutation. This is significantly higher than if you look at events between civilians. And then interestingly, if you look at the events that go from prisoners to civilian, this proportion is almost 100%, so an overrepresentation, suggesting that somehow these spillover events, or just generally TB transmission events among prisoners, these compensatory mutations somehow are overrepresented in these transmission events. So this is our current hypothesis that perhaps during this incarceration, the way that how TB evolves within these prisons somehow creates hypertransmissible multi-drug resistant strains. This is our current hypothesis. And again, this was a retrospective study. We went then ahead, as I mentioned, essentially we collected all TB strains we could get from the whole country, and then we selectively started to sequence them. So we started with MDR strains again, MDR plus, so pre-XDR and XDRs. Now these are the following 3 years, 2014, 15, and 16. So this time we had a much higher coverage because we were prospectively collecting and sequencing 992 strains, about 95% of all reported case-confirmed, culture-confirmed MDR cases. First thing we did, then we pooled all the MDR data we had of the 6 years. So it's about 1,600 genomes shown here, 2011 to 16. And what is actually striking, what comes out is actually, if you look at the proportion of the pre-XDR and XDR, it's actually almost 2/3. So essentially that means that the so-called MDR epidemic in the country of Georgia is actually a pre-XDR/XDR epidemic, obviously showing that it is a big problem. Now if you look at the flavor, if you will, of pre-XDR, it's also interesting to note that the majority of these so-called pre-XDR strains are actually MDR plus injectable resistance. And then, of course, we have quite a lot of XDR strains also transmitting. We also looked at some old drugs, streptomycin. 94% of all these strains had a resistance to streptomycin, maybe not surprising, but it's essentially almost fixed. The whole population is essentially resistant to streptomycin. We started to look at the new drugs. Clearly, this is difficult because there's very little known about resistance. Nevertheless, there's instances of non-synonymous mutations in genes that have been associated to resistance to bedaquiline and delaminate. We know bedaquiline has been used in Georgia since 2013. Delaminate is only recently been introduced, so we don't really know what these mutations are doing. This needs to be confirmed with DST. Going back to the compensatory mutations, we then looked at the association, potential association with different resistant profiles, and perhaps not surprisingly, we see that our compensatory mutations were also associated with pre-XDR and XDR TB. So suggesting that these compensatory mutations not only help or contribute to transmission of MDR, but also facilitate the acquisition of additional resistance and then the onwards transmission of pre-XDR and XDR. Okay, but now actually, what we really wanted to do, the reason we went ahead was actually to compare to pan-susceptible strains, because at the end of the day, that was really the question. So does MDR strains transmit less, equally, or more than pan-susceptible strains? I said we had collected all TB strains in this period of time. So now we go back to 2014 to 16 to be fair, right, in terms of the comparison. We sequenced all pan-susceptible strains, so about 3,000. There's another about 1,000 strains we still have in the freezer, which we chose not to look at yet, because they are have all kinds of different variants of monoresistance, polyresistance, etc., which would make things complicated. So we said, let's start just comparing MDR to fully susceptible strains. So what we see first is so here is a tree with all MDRs and susceptibles, roughly 4,000 genomes. The striking thing, lineage distribution, I referred to this, MDRs, 80% of MDR strains are lineage 2, so this I already referred to. We kind of expected that, but not to that extent, really. If you now look at pan-susceptibles, it's almost the mirror image. Again, it may be not surprising to some of you. I wouldn't have expected that extreme difference. So this was one aspect. But now let's go to the actual question, which is the transmission. So again, using clustering as a proxy. So this is now the second 3 years, 14, 15, 16, we essentially see the same thing. Kind of a hypothesis confirmed, right? So compensated multi-drug resistant strains transmit more than regular MDR strains. So that was nice to see, replicated in the following years. Now, what we hadn't expected, well, me definitely didn't expect, pan-susceptibles. A priori, no difference with MDR, and the compensated MDRs are essentially superbugs. That's what we'd interpret from this. But, of course, I just referred to the fact that we have these very big lineage differences, so we had to look at this stratified by lineage. So when you do this, you see the following with lineage 4, which is actually what I had expected. There's a slight trend that you go from MDR, which is least transmissible, to compensated MDR, slightly more transmissible, to pan-susceptible, which is yet more transmissible. Now, with this analysis, there's actually no statistical difference between these different groups. Now, for the genomic epi-aficionados, we actually use another proxy or another measure of transmission, which is the terminal branch length, which is another way to actually look at this. So the shorter your terminal branch length, desto more transmission. And when you actually do the same analysis, we see essentially the same picture, but we actually do have a statistically significant difference between MDR and pan-susceptibles. But the difference is not very big, right? It's a small difference. But this is actually not super exciting compared to what happens with lineage 2, which is shown here. So lineage 2 really, again, jumps out as being very different. And note that essentially all lineage 2 strains are more transmissible regardless of resistance profile, regardless of being compensated or not. That's compared to lineage 4. We have a very big difference between MDR and compensated MDR in lineage 2. The fact much more extreme than what we see in lineage 4. And essentially we see zero difference between compensated MDR and pan-susceptibles. What we haven't done, I have to say, okay, here also we apply the other measure, the terminal branch length, and we see essentially the same effect. A lot of this is preliminary. We essentially got these sequence data just a few days ago. I promised last year we'd actually talk about this if I would be invited again, so that's what I wanted to show this. We need to do more analysis, of course. The immediate question comes, okay, what about prisons, right? How do prisons now play into pan-susceptibles as opposed to MDR and things? So this we haven't done yet. So this is upcoming in terms of the analyses. So in terms of conclusions, I think it's pretty clear that compensable mutations are not just interesting and important in the lab, but I think they clearly also play a role in epidemiological settings because they're associated with transmission of MDR, XDR, TB. They're overrepresented in these prisons to civilian spillovers. There we need to do more work, but there is this idea that imprisonment might actually cause an intensifying drive for the evolution of highly compensated multi-drug resistant strains, which then perhaps could easily spill over into the general population. These compensable mutations are associated with increased resistance, pre-XDR and beyond. We have this interaction between lineage 2 and compensable mutations, so that's, I guess, the perfect storm, if you will, or the superbug is a lineage 2 strain which is highly resistant, has a compensable mutation. It's not just any lineage 2, I didn't refer to. If you looked at carefully at the tree, you see there's actually very little diversity in the lineage 2 tree compared to the lineage 4. So there's clearly a sub-clone or variant of lineage 2, which somehow were created in that part of the world, which seems to be extremely successful. And as a result, essentially compensated MDR, XDR TB at least can be as transmissible as pan-susceptible TB. And with this, I would like to thank many collaborators, other members of the lab, funding agencies, and you all for your attention. Thank you very much.
Thank you very much, Sebastian. The floor is open for discussion. I already see hand waving by Marcel Beer in the back. Yeah, you were first. At least you're the one standing, so you're easily picked out of the crowd.
Hi. Question from Simon Grandjean. What's your threshold for calling a cluster? If I understood, you have 95% of MBR in the country, and only 40% are clustered. So where are the other 60% coming from?
Yes, so I forgot to mention that. So we have used a very conservative clustering measure, which essentially we had to at least 3 members, as opposed to 2. Most people use 2. We use 3. We use the traditional 5 SNP threshold for this analysis, but as I mentioned, we also use this terminal branch length analysis, and we're going to do more.
Yeah, I think the gentleman over there was first, and then after Ed.
Thank you very much for the talk. I'm Dr. Venkat from Apollo, Hyderabad. So in your talk, you mentioned the so-called pre-XDR strains have a high proportion of aminoglycoside resistance compared to fluoroquinolone, but in India, we've observed the reverse, where because of the excess use of fluoroquinolones in the community. Do you think that is linked to aminoglycoside use in the community?
Yes, exactly. That's a very good point. I wanted to mention this. This clearly, presumably, has to have to do with the history of particularly kanamycin use. I mean, there are anecdotes I hear from people in Georgia that they say, okay, you get your regular TB treatment, but make sure you get an injection, to make sure that only real injected drugs are real drugs. So presumably this reflects clearly the past history of TB treatment.
Ed?
Thanks, Sebastian. You alluded to this, but I want to emphasize it. The requirement for transmission, of course, means that you would lose a lot of non-transmissible strains. So there's always this pressure to maintain those strains that are fit enough to transmit. But the question is, in the prison, as you alluded, is there particularly easy to transmit, and therefore facilitating this? And then we're totally ignoring the host. Are there subpopulations, the healthier populations, that are able to transmit, only able to transmit susceptible disease or receive susceptible disease, and are sickle HIV infected, for example, persons more likely? So there's that dimension that we assume evens out, but we don't know that, do we?
I agree, totally. So HIV is very low in Georgia, fortunately, but there may be other factors, alcoholism, and people with prisons obviously have many other problems. This idea that maybe counterintuitive, that yes, transmission in prison should be easy generally because we have so many people. But the idea is that really through this ongoing transmission, something happens to the bacteria. There's a lot of replication. And perhaps that's, again, as I mentioned, maybe the way how these compensatory mutations are kind of selected for.
Yes. One more question. I think Emily was first. Yeah.
Hi, Emily Kendall. Question about how much of the difference in clustering may be due to factors other than sort of intrinsic transmissibility. Is there any way to think about how much of this is due to particularly differences in the infectious period? I don't know how much rifampin sort of susceptibility testing there was in Georgia in 2014. I assume it wasn't perfect up front and that there may have been delays in diagnosis and appropriate treatment that lead to increased transmission.
I don't know. But at DST was done. I mean, that's one reason we could go there, because it's a very centralized system, so they collect all the strains, everything is sent to the central lab, and they have been doing standard DST, at least before 2011. That's also the reason they stored only the resistant strains and not all the strains. So that's why we could actually go back. So at the minimum, we know that you're actually resistant strain. Now, in terms of treatment, yes, treatment history is extremely difficult to reconstruct, particularly in the retrospective study.
Thank you, Sebastian. This was great. Very interesting. I have a question related to Ed and Emily's, actually. I think it's really important that when we look at transmissibility, before we blame the bug and say it's a super bacteria, because obviously the repercussions of that are potentially maybe public health practitioners interpret that this is the bacterial fault, maybe we shouldn't intervene like on a public health level to the same extent, or we're fighting a losing battle of some sort. I think the implications of blaming the bacteria are potentially larger than one would imagine. So I think it's important that we, for example, measure like smear positivity and look at contact exposure, duration of exposure, things like that, and control for these factors. So I'm really quite curious to know, once you look at controlling for imprisonment in your model, whether that actually your observations are robust.
Yeah, no, I agree. So this we haven't done yet. I mean, it seemed to be robust in the retrospective part. Now, of course, we only looked at resistant strains. But yeah, totally take your point. Absolutely.
Okay. Thanks very much, Sebastian.
And just to remind you, at the end, we still also have a few minutes for questions. Now it's my pleasure to introduce Professor Annelies van Rie, who's professor in epidemiology and social medicine at the University of Antwerp. Welkom, Annelies.
Good afternoon. Everybody had a Fit To, and so while I was sitting there, I thought I don't have a Fit To title, so I'll change it to Fit to Fight, and you'll see why. But the title in the book is Tolerance in Mycobacterium tuberculosis under drug pressure, and I wanted to upfront acknowledge Sander Goossens, a PhD student in my group, who has helped me tremendously in digging through the literature. Being a clinician, I like to always start from a clinical point of view, and I've always been fascinated by the observation of great interpatient variability in response to treatment, even among patients with proven pan-susceptible TB who are fully adherent to treatment. And this variability shows itself in differences in time to culture conversion, with some people culture conversion after a couple of days and others being culture positive for weeks. And some patients relapse in the absence of acquisition of drug resistance, and most don't. Predominantly, interpatient variability has been the causal factor or the attributed to host factors, being the baseline mycobacterial load, presence of cavitary lesions, presence of HIV infection, and a smoking history in the patient. But I was wondering, does mycobacterial tolerance contribute to this interpatient variability in treatment response? And there's definitely experimental data to support this type of different phenotypes in MTB that have different growth characteristics in animal models. So time to kill curves of MTB under drug pressure are typically biphasic, and sometimes people have found multiphasic curves. Here you see, this works. Okay, so you see growth and then a rapid decline when the culture is treated with isoniazid, and then a change in kill curve. So what Ahmad et al. Showed that this was not due to selection of INH, because this was INH mono treatment, but due to the selection of phenotypically tolerant persisters. In human models, using PK modeling of changes in time to culture conversion of serial clinical isolates collected from patients, these data suggested that at least 2 and probably 3 bacterial populations with different growth dynamics and kill rates were present. And so I moved further on these 3 MTB phenotypes with different growth characteristics in these human models. And looking at fully or highly susceptible, a second group tolerant, and a third group persisters. And as a working definition, use the definition published by Mattern in 2018, applicable to other bacteria, which states that tolerance is a temporal and reversible ability of bacteria to survive exposure to bactericidal antibiotics. And if you take this type of approach, then tolerance is broader and encompasses persisters. So how to think about these 3 different MTB phenotypes? Well, if you look at the fully or highly susceptible, which aren't just drug susceptible, they also have a susceptible phenotype. In patients whose MTB population would be purely comprised of susceptible phenotype, you have very fast culture conversion, you have 0 relapse rate, the MIC is below the ECOF, and the MDK, which is the median days required to kill a predefined percentage, which can be 50, 75, 90, 95 of the bacterial population upon drug exposure will be low. You don't need that many days. If you look at tolerant persistence, which has been studied a lot, you have still fast culture conversion because the persisters are always only a small proportion of your bacterial population. And therefore, you have fast culture conversion, but because these persisters hang around, you actually have an increased relapse rate. Your MIC is still below the ECOF, which is different from the drug-resistant population, and your MDK, because you have fast culture conversion, your MDK is also low. If we look at the other form of tolerance, there's actually slow culture conversion, and that's typical, explaining this interpatient variability in time to culture conversion. The relapse rate, however, is 0 or very low compared to drug-resistant TB and populations that contain persisters. Your MIC will be below or around the ECOF. People have talked about creeping MICs, coming a little bit higher towards the ECOF or just around or above. And the MDK, because culture conversion is slow, the MDK will be increased. So how do the existence of these 3 MTB phenotypes in a patient now relate to microbiological treatment outcomes? And Nicholas already this morning used the comparison with soldiers, and surprisingly, I also looked at war. I don't know why we both did that, but there must be something there. So we have the enemy, and I went back to the Roman times. And so in the Roman times, you had 2 types of soldiers, or probably more, but 2 main types of soldiers. You have the low socioeconomic class, poor soldiers who were not really trained and were just kind of thrown in there to be killed. Then you had the higher socioeconomic class soldiers who actually went through military training, and they were equipped to fight. And then I jumped to World War I, and there you have the people in the trenches. So if I look at that, the enemy in TB is the antibiotics. We have our fully susceptible. They're not really equipped. They just get killed. Then you have your tolerant, who have some ability to sustain this drug pressure. And then you have the coward persisters who hide and make sure that they're not seen, and they sit in the trenches. Now, what happens when we have a peace treaty? When we have a peace treaty, normally the population that is attacked is not totally decimated and disappeared, so there's still some people left. So if we have a peace treaty here, we would, the susceptibles, they're gone, they're died. Some of those tolerant, of course, also didn't survive the attack, but there's still some alive. And those that were, that hid in the trenches, they now jubilate and come out because the enemy disappears, the susceptibles have disappeared, and now everybody else comes out. Unfortunately, what we know with history is one conflict often doesn't end and leads to a new conflict. So what happens then? We have the enemy comes back. And And we have persisters, but the persisters they take about the same population, the same proportion of your new army, because these persisters are actually a phenotype, the characteristics of humans. They're the cowards who hide, right? And this is just a phenotype. This has nothing to do with the fact that there was a war before. Whereas the rest of the population, the army, has now understood what the tactics of the enemy are. So they come back, and now they have adapted techniques to actually fight. They're fit to fight. They're able to fight the enemy better. And so we have less of these fully susceptible. So let's go back to science. And so what happens? If we have these susceptible population, right? You have a fully highly susceptible population. You add antibiotics, everything is killed. You then take the antibiotics away, and you have no regrowth. If we have persisters, then we have this mix of fully susceptible with a few persisters. You give antibiotics, those that are highly susceptible get killed, your persisters stay. You take your antibiotics prematurely away. What happens is you actually regrowth of persisters, but predominantly, again, fully susceptible, highly susceptible, with the same proportion as persisters as before. The other tolerants, we have fully susceptible now with some tolerant. Some of the tolerants get killed, so you have few left, but when you take the antibiotic away, there's regrowth, and now they're all tolerant. And with what happens with resistance, we all know. So my goal was to investigate the tolerance from a clinical perspective, using clinical data and a multi-omics approach. To start with, we chose to go for genomics, transcriptomics, and epigenetics. We start with a retrospective study, use stored serial cultural isolates from RIF-resistant TB patients, and to identify slow and fast responders. This is just a subselection of the patients that we have samples from. And as you can see, for example, this person here has 2 positive cultures and then a whole slur of negative cultures, and so very rapidly becomes culture negative. The same person here, only a few days positive and then negative. In contrast, here you have people who, after 80 days, still never have a negative culture. The negative cultures are in the little dots. I don't know where I am. Okay, the dots are negative cultures, the triangles are positive cultures. So, the hypothesis would be that in those patients who very fast culture convert, that we don't find signatures of tolerance, whereas in the patients who prolonged have culture positivity, that we would find and persisters and tolerant phenotype. Then we will also do a prospective study where we will not only look at cultures but also at sputa, and do serial multi-omic studies on these isolates of patients who were in a clinical trial. I had hoped that I would be able to present some of the retrospective data, but the analysis, as often in research, is a bit delayed. And so what I will focus on is what we know from literature, and hopefully next year or later I'll be able to present more real data. So what is actually known about the mechanisms that MTB employs to become tolerant? So there's mechanisms for general drug tolerance, and then for almost every drug there are some specific mechanisms used by these drugs. So there is transcriptional regulation, metabolic shifting, cell wall thickening, upregulation of efflux pumps, and acquisition of genetic variants. These don't work in isolation. So you have transcriptional regulation that leads to metabolic shifting and so on. So transcriptional regulators really coordinate the drug tolerance, and YB transcriptomic factors and sigma factors are the 2 most described. We have the YB7 regulin, we have the YB3 regulin, and we have the SIG-A factor. YB7 has then different genes. YB3 works on redox sensor protein, and SIG-A is interesting because it actually forms under stress a promoter complex with YB7 and SIG-A, and then interacts with the dormancy regulon. What you can see here, so we have the interaction of YB7 with SIG-A and then SIG-A with DUS-R. We look at metabolic shifting. Most antibiotics target metabolically active bacteria, so lowering your metabolic activity and reducing growth is a way to evade drug pressure. And this experimental evidence of upon treatment there is reduced protein synthesis. There are main pathways involved in energy metabolism, such as the aerobic respiration and ATP that are down-regulated upon treatment. And there's some pathway reshuffling, especially a shift from the use of carbon via the growth-promoting TCA cycle to storage of carbon in fatty acids through TAC synthesis. And so here you see that you have the TCA cycle, which mainly leads to amino acid synthesis, and if you move away from that, you go to fatty acid. This is, of course, also described in dormancy, so this shows that there's overlap between persistence and dormancy. Cell wall thickening, you have the hydrophilic drugs that use porin systems and the lipophilic drugs that come through with diffusion. If you have stress, then your cell wall will thicken, which has a consequence that the diffusion is more difficult, but also that those porins are much more difficult to access. So for both types of drugs, you have problems, you create problems. Interestingly, the cell wall thickening increases from pan-susceptible to MDR to XDR. This is probably not related to the mutations conferring resistance, but to tolerance that precedes or accompanies the acquisition of drug resistance. We all know about efflux pumps. This is the opposite of the porins. Here now you pump the drug out. And if you have fixed mutations, then it's not transient, so you don't call it a tolerance, but it's actually resistance. But if it's upregulation under conditions under a mechanism that is condition-dependent, reversible, and transient, then it is tolerance. And of course, these efflux pumps can be specific for one drug or can affect multiple drugs. When you think about this concept of tolerance that being transient, you would say, well, genomic variation cannot really play a role. However, there's three interesting examples. There's the PRPR gene variant, which is selected for when a drug is, when MTB is exposed to RIF, INH, and ofloxacin. And this alters the propionyl coenzyme A metabolism, which leads to drug tolerance. But this tolerance is only a specific on the condition because it only occurs in propionate supplemented media in absence of vitamin B12, and it's reversible because it's resolved upon addition of vitamin B12, and so it fulfills the criteria of tolerance. There's what they call the highly persisting, and that's one of the complexity of this literature review that we were doing. It's tolerance, but they call them highly persistent, and you have tolerating persistence and persister tolerators, and everybody mixes everything, but so highly persisting here relates to tolerance. The FAD-E30 mutants that are isolated in MTB surviving RIF and streptomycin pressure. And so this FAD-E30 is involved in lipid catabolism, and which is then again related to this metabolic shifting. And then there is the transient frame shift mutations in the GPLK gene, and that gene is involved in the promotion of the stress response regulators, DOS-R3, and some SIG factors. And the authors conclude the adaptive mechanism that mediates its reversible on-off switching of a gene could include DNA methylation, homologous recombination, DNA rearrangement, or insertion deletions in short sequence repeats, or homopolymeric tracts located within the promoter region or the codon sequence of a gene. So it's a bit different than the fixed mutations that we traditionally think about with resistance. I'll just quickly give one example of drug tolerance and used bedaquiline. So upon bedaquiline exposure, MTB is pushed into a tolerant state. It activates transcription factors, increases expression of the ATP synthesis, upregulates RV032 targets, and again, that metabolic shifting. What I found extremely interesting is that the authors found that the development of bedaquiline tolerance may be inhibited by combining bedaquiline with protonamide. So why care about drug tolerance? Well, it leads to reduced killing. So we have prolonged infection, increases lung damage, as we saw this morning, increases risk of acquisition of resistance. And related to this session, you have prolonged infectiousness and increased transmission. So prevention of the development of tolerance, of the reversal of already existing tolerance, could lead to development of novel treatment shortening strategies. So is this pure fantasy, or can we actually do this? So there was one interesting recent publication, Enhanced Respiration Prevents Drug Tolerance and Drug Resistance in Mycobacteria Tuberculosis. I'll quickly, because I'm running over time. So here they added cysteine to INH. Cysteine on itself had no effect whatsoever, but if you do INH alone, you have kill and then recovery. If you add cysteine, you get persistent kill and actually close to sterilization. So this is an example of cysteine, of a tolerance attack mode. But interestingly, you had to give the cysteine on the same day you started INH or within 1 day afterwards. It had no effect anymore. And they tried other components, but you had to have a free trial group. And how it worked was, again, transcription with upregulation of DOS-R, DNA damage, and cation homeostasis. So multiple phenotypes of MTB exist. In addition to those highly susceptible phenotypes, the presence of persisters is well established, but I believe there is evidence that there is a third kind of tolerant, and it seems possible that we can develop drugs targeting tolerance, and that this would be a useful addition to what we currently do. And then I would like to thank these people who gave very important input and samples for the future studies. Thank you.
Thank you very much, Annelies. We have time for one question.
Hi, Anis. Thanks for the nice talk. I am Vijay from Oxford University. I wanted to know whether mutation rate, is there any difference between tolerance and susceptible or persisters?
It's a very interesting question, and I hope that I'll be able to answer that when we've done the studies. There's all the tolerance studies to date have been done in models, in animal models and in vitro models, and so I think we really would need some human studies to be able to answer this mutation rate, because we're interested in mutations that happen within the host.
Thank you, Annelies, and thank you to all the speakers. So please come back at 3:30 after coffee break, and as Frank requested, stop talking loudly if you don't come back.
May I now invite Annelies Messman. She is a postdoctoral fellow at the Harvard Medical School Department of Global Health and Social Medicine. She has done her PhD from Experimental Immunology Lab from University of Amsterdam, and has been previously involved in leading lab programs for partners in health Sierra Leone. Annelies. Okay, just wait for 2 minutes. Yeah. Let people settle down. Okay.
Take a sip of water that's untouched. Oh.
We have 15 minutes. Yep. And the lady there will show you 5 minutes left or 2 minutes left.
Sounds good.
Oh, I thought this was the panel. Sorry.
Okay, Annelies, please start.
Good afternoon, everyone, and thank you for staying in this TBScience session. And thank you also for giving me the opportunity here to talk about differentially culturable TB. So, to start with a brief introduction, the two main methods to microbiologically detect live bacteria are smear microscopy and culture. And in most settings where TB is highly incidence, screening and diagnostic tests are usually done with smear microscopy, which is not very sensitive, while culture, which is like the gold standard, very high sensitive test, is not always available. Yet, there's still a high percentage of patients that have culture-negative TB, meaning that they are diagnosed with culture-negative sputum samples and they have pulmonary TB. And this is generally thought to be related to either early disease or a low disease burden, for instance, in patients with HIV. It also happens sometimes that samples are smear positive but culture negative, and this is often thought to be related to the presence of dead bacilli or errors in the lab. There are, however, several lines of evidence showing that not all the TB bacilli are able to grow in standard media. And I just wanted to point out a few work of a few groups who are working on this. And this includes the work of Bhavesh Khanna, who came up with the term differentially culturable cells. And their group showed that some subpopulations of cells only grow in the presence of culture filtrates, so there's probably a secreted factor in culture filtrate that enhances the growth of these cells. And then a group of Carl Nathan, they reported similar results, as well as they can induce this differentially culturable phenotype by starvation or under drug pressure. And then there's a different type of work, but also relevant for this. Recently, two groups, including the group of Alexander Pim, they showed that genetic diversity in sputum is much higher before compared to after culturing a midget. So this suggests that midget culturing selects for subpopulations of cells to grow. But not much is known about this phenomenon of what these cells are. So for the present study, our hypothesis was that maybe carbon source availability in culture media affects this culturability of these cells. And the reason we had this hypothesis was because the carbon source in culture media is different maybe from the carbon source in the hosts. So in culture media, the main carbon source is glycerol, which is added to the media because it can lead to a rapid growth of the bacilli. But in the lungs, it's known that lipids are an important carbon source for replication and pathogenesis. And to investigate this, we made use of a biobank of samples from a large EPI study that was conducted in Peru between 2009 and 2012, so quite a while back. And in this study, 4,500 index patients and household contacts were recruited. And from everyone who was diagnosed with TB, a sputum sample was collected at enrollment after 2 months, and in case of drug-resistant TB, also at later time points. And this sample was tested with microscopy by culture. It was grown on algae slants, and part of the sputum was also frozen at -80, so this allowed us to use it for the present study. So we went back to these records from this study, to the lab results, and purely based on the lab results, we selected 15 samples that were highly smear positive, so they were AFB 3+, but they were negative in LJ culture. And it turned out that these samples they represented a mix of samples that were collected at baseline, at enrollment, or at later time points after treatment. It also was a mix of several strains. It was a mix of samples from index patients or their household contacts, and some of these patients had been diagnosed with drug resistance and others with drug susceptible TB. So our study setup was the following. So from these 15 samples, we aimed to grow them either in a medium with glycerol but low on lipids, so it's the 7H9 with glycerol but no oleic acids, or in this house-made lipid broth that had no glycerol but had a high concentration of fatty acids, monodi, and triacylglycerides. And if these cells would grow, we would investigate their phenotype by looking at microscopy, do a glycerol toxicity assay, as well as conduct whole genome sequencing and look for drug resistance in MICs. So interestingly, 10 of these 15 samples were able to grow, that were not able to grow before on LJ, were able to grow in this lipid media. And 2 of these 10 also grew in glycerol 7H9. All of these 10 lipid-grown cultures were AFB positive. And while only 2 of these samples directly from sputum grew in glycerol, after their initial growth in the lipid media, 9 of these 10 could grow again. So they could revert to the utilization of glycerol, which is shown here. These are the growth curves of these 10 samples. And although the kinetics are different, 9 of these 10 at least are able to use glycerol again for their growth. So next we want to see if there's any mutations that cause this phenotype. And first we assessed for mutations in a gene that was also introduced in the previous talk, GLIBK, which encodes for glycerol kinase and is essential for the utilization of glycerol. And this gene, it has been shown that there's a lot of samples or a lot of bacteria can have a transient mutation in the promoter region of this gene. But in our sample set, only 1 sample had a frameshift mutation caused by the insertion of 1 nucleotide. So this mutation in this sample might have contributed this effect, but it was not the cause for all these samples. So to find novel mutations that could be related to this phenotype, we next compared the sequences of these 10 samples to a reference strain, and we used 2 different approaches. So 3 of these samples were collected from patients that had been on treatment for a long time, so at least over a year, and that had been culture positive at baseline. So we had their baseline sequence, we had this lipid-grown sample that was collected later, so we could compare these 2 over time. And we found with this approach that 16 SNPs. None of these SNPs were present in more than 1 sample. In the remaining 7 samples that from these patients that had only culture negative samples, so we could not do this approach, but we went to our database of almost 2,900 sequences, all from Peru, and we found their closest genetic match. And this led to the identification of 47 additional SNPs. And 41 of these 47 SNPs was shared by 3 almost identical isolates, and these were from 3 patients that were not related to each other. So there might be some genetic component here, but again, we could not find like a single specific gene or pathway that was shared by all these 10 samples. So the last thing we did was look at drug resistance, and first by whole genome sequencing, we found that 6 of these 10 samples contained SNPs that are associated with drug resistance. 3 of these patients had been diagnosed with MDR, so this confirmed their status. 3 other patients only had culture negative samples, so this shows that this culture negative phenotype actually prohibited the diagnosis of MDR. And then we looked at phenotypic resistance, and strikingly what we saw is that all these 10 samples, and what is shown here in the black bars, we did this in 2 ways, the MICs. We did this in the lipid media and in the glycerol media. And here in the first bar is shown the result from our control, H37RV. And what you can see is that all these samples are highly resistant to isoniazid and ethambutol. And this is not related to the presence of any drug resistance associated SNPs. And even when we transferred these cells to the glycerol media, this drug resistance was maintained. And this effect was specific for these 2 drugs, because if we look at rifampin, then we see that only the isolates with a RPOB mutations were resistant against rifampin in either of those culture conditions. And for streptomycin, also all the isolates were resistant in the lipid media, but when we transferred them to the glycerol media, only the samples here with those red asterisks that had these mutations remained drug resistant. So to summarize these findings, what I showed here is that some smear-positive samples grow on lipids, but do not grow on standard glycerol-rich media. We could not find a single gene mutation or a single pathway that is related to this phenotype. And the carbon source is very important for phenotypic drug resistance, we think, because all these samples were very highly resistant to several drugs. And the variation in some of the results suggests that there might be multiple mechanisms leading to this phenotype, but we hope to find it out in future studies. I just would like to acknowledge some people who were involved in this study, and particularly Sung-Hoon Baek in Korea, who did a lot of the lipid culture work, Tom Yerger and Chuan Chin, who were very helpful in the whole genome sequencing analysis. And this was done in the lab of Megan Murray and all funded by NIH. I'm happy to take any questions.
Thank you, Annalise. Any questions?
Thank you. Very interesting. I wanted to ask you, is the conclusion that these isolates are only phenotypically resistant in lipid media? Because I think you showed, I know that the MICs were different, but they both seem to be high. Is that not true?
Yes, the reason for isoniazids and ethambutol, it's true that in the glycerol media they were also resistant to it. The reason why we're a bit cautious now in our conclusion is because we don't know what the effect is for patients, because some of these patients received isoniazid and they seem to be cured. So, yeah.
Hi, Marcel Baer from McGill. It's been my impression, by the way, great talk. It's been my impression that the culture negative is most common in extrapulmonary samples. Have you done the glycerol versus lipid culture on any extrapulmonary biopsies or anything like that?
We have not. It would be very interesting to do, but this was only pulmonary TB, yeah.
Sorry, can I ask one more question?
Sure, go ahead.
Did you, there might be some stochasticity to growth, right? Is it possible that if you had taken the sputum and replated them just on regular media, that they would have grown, or did you guys do that?
They did not, I mean, the fact that they did not, this was obviously like a small study, and a lot of things we want to, we need to confirm with more samples and more culture media, types of media. But I think that we show that they do not grow at all on LJ, which is, incubated for quite a long time. They do not grow on standard midget for 3 weeks, but then they do grow in the lipid. I think that's, for now, enough evidence to continue this, yeah.
No further questions? Thanks, Annalise.
So the next speaker will be Chuan-Ching Wang, who is with the Department of Global Health and Social Medicine at Harvard Medical School. The floor is yours.
Good afternoon. My name is Chuan-Ching Huang. Thank you for giving me this opportunity to share what our group have done. So the topic I'm going to speak on today is a friendship mutation in the GLPK gene, so glycerol kinase gene, and the drug-resistant phenotype of the MTB. So recently, 2 papers being published about the GLPK and the MTB. So the first one was done by our group and Dr. Chris Satti's group. We found that a common variant in the GLPK gene reduced the tuberculosis drug efficacy. And almost at the same time, Dr. Allen's group published a paper saying that a phase variation in the GLPK gene of the MTB produces a transitionally heritable drug tolerance. And so the result I'm going to show you today is kind of an extended analysis based on the findings of these 2 papers. So I'm going to give you a brief introduction about the GLPK gene and the variant that we will focus today. And this actually been covered by several of the previous presentations. So this is the carbon metabolic pathways of MTB. And the MTB is a bug that is capable of using lipid or the glycerol as the carbon source. And the GLPK play an essential role in the glycerol pathway. And So it's catalyzed the phosphorylation of glycerol and yield the glycerol 3P. And there is a 7 base pair homopolymeric cytosines in the GOPK gene. And the variant that we will focus on today is insertion of a C, so from 7 cytosine to 8 cytosine. And this insertion result in a frame shift and lead to a downstream stop codon. So isolated with this frame shift, the translation of a GOPK gene will be incomplete. So what we have known about the GOPK variant and the MTB. So first, using an in vivo model, Dr. Chris' team shows that a knockout GOPK strain actually has an almost identical survival curve as the wild type. So in the vivo settings, the knockout GOPK gene does not seem to affect the fitness of the bug. However, when the drug is presented, the story is different. So Dr. Allen's team shows that, as you see here, the orange line is the GOPK knockout strength, and it's increased the tolerance when the drug is presented compared to the wild type strain or GOPK knockout plus the complementary. And using and also Dr. Allen's group described the phenotype of this frame shift mutation as a small colony phenotype. So as you can see here, the first one, the bottom of the first row, you can see there are some colonies much smaller than others, and those are the isolated with a frame shift mutation. And the second, Dr. Allen's team also found that this frame shift mutation seems acquired by the TB bug rapidly. So the middle and the bottom plate is done a week after the application of the drug, and as we see, it starts showing some small colony. And in addition, this phenotype seems is reversible. So this is this one, the right-hand up dish is a passage 4 of an isolated with frame shift mutation. And if we remove the selection pressure of drug, then at the passage 11, we start seeing some isolated growing bigger, suggesting a reverse reversion. So in a clinical isolate, our team found that this frame shift mutation appears multiple in a phylogenetic tree, suggesting that this mutation is a general mutation and it have multiple origins. So it's not lineage specific. And also we found that almost all the isolated with GOPK frame shift mutation are resistant to multiple drugs. And as you see in the right-hand side figure, the dark star is the isolated with the GOPK frame shift mutation. And as you see, almost all of them are red, meaning it's resistant to more than 7 drugs. So based on this, I want to introduce my hypothesis. So I think a TBScience person likes soldiers a lot, huh? Yeah. So this is a soldier, and a soldier will need to have an enemy, right? So when the enemy comes, a soldier is capable to acquire a shield rapidly, and then increase its defensive power, so it can survive. And this extended survival increase the likelihood of the soldier to obtain a sword. Okay, so when we look on these survivors, those with a shield are more likely to obtain a sword. So let's replace the soldier by the TB bug. So the TB bug, when the TB bug is under drug pressures, it can develop a rapid GOPK frame shift mutation rapidly, and increase the drug tolerance, then extended the survival, and wait for the occurrence of the causal mutation. And as long as they have obtained a causal mutation, they become drug resistant and they can survive. So when we retrospectively, so we cross-sectionally see those resistant strains, we hypothesize that among the drug resistant strains, clinical isolated with the GOPK frame shift mutation are more likely to have an extreme minimal inhibitor concentration, as we know that usually the causal mutation result in the high or extreme MICs. So the study isolated of this analysis are from two sources. One is the about 2,500 isolated from a core study conducted in Lima, Peru between 2008 and 2012. In that core study, we identified incident TB cases as index cases, and we enrolled their household contacts. So in total, we enrolled 4,500 index cases and 14,000 household contacts. And we followed index cases for the treatment outcomes, and we followed household contact for the TSC conversion or the secondary cases, incident cases. And the second source is stores samples in the Massachusetts Pro-National TB Reference Laboratory, and most of the strain over there are MDR cases. So we have about 600 isolated from that storage. So in total, we have about 3,100 isolates. So this isolate underwent the whole genome sequencing, and about 200 were low quality or mixed strains. And because, as I said, the analysis is conditional on the resistant pattern, so we exclude those who were drug sensitive and keep those at least resistant to one first-line drug. And among them, we have about 1,000 isolated with MICs, and among them, we identify about 150 with GOPK French mutations. And almost all of them were NDR, compared to about 75% were NDR among those without GOPK French shift. And the cut of the extremely MIC we use is showing in this table. And the right-hand column is the folded change compared to the WHO cutoff for the definition of the resistant. So these are the MIC, extreme MIC cutoff we used. So here's a result table. So among the so I want to emphasize again, this analysis is conditional on the resistant strains. So, for example, the IHR rows, you see the strains with GOPK mutation has 83% with extreme MIC compared to lost wild type. Wild type GOPK has 70%, and it's significant. And this increased extreme resistant pattern occurred in the French shift GOPK mutation strains to INH, to RIF, and to streptomycin. And after p-value, as I show here, and we further adjusted for the population structure of these isolates using a genetic relativeness matrix. So after the adjustment, the association between the GOPK French mutation and the INH and the RIF drugs remain strong. So the conclusion is isolated with the French mutation are more likely to have extreme resistance to INH or RIF. And so the GOPK mutation seems to provide an intermediate stage for NTB survival before the occurrence of the causal mutation. So some future step is we want to evaluate whether these strains with GOPK mutation really have the known causal variants. And we'll also verify the result using an independent data set. Thank you very much for your attention. And thank you for my advisor, Dr. Megan Murray, and all the fundings and all the help from all the colleagues. Thank you. Thank you.
Thank you. The presentation is open for discussion. Yeah, question in the back.
Very nice work. Did I understand on the table at the second last slide that you saw GLP as associated with rifampin resistance but not rifabutin resistance? And is that what the RUB is?
Yeah, but we have a much, I think we have much less RBU first resistant, and the second is, I think it's also, you still see a signal, but it's not significant, right? Yeah,
okay. Okay. Yes. I wasn't sure if it was a numbers thing or if there was a discordant observation. Sorry, because I just came through. Thanks.
Yes.
Are there any more questions? If not, thank you very much. And we'll move on to the next speaker.
And then it's my pleasure to introduce Catherine Walter, a postdoc at Stanford University, who will explain how pathogen evolution can inform our understanding of epidemiological dynamics.
Thank you for having me. I'm going to speak today about some of our work investigating how we can leverage increasingly available genomic data to better understand tuberculosis transmission. Mycobacterium tuberculosis is constantly evolving within individual hosts over the course of infection, between hosts over the course of the transmission process, and as the bacteria spreads over wider geographic areas. And this constant evolution can create a powerful epidemiological record. We don't observe transmission, of course, and there are many outstanding questions about where transmission occurs and between whom, particularly in high incidence areas. The accumulation of mutations over time, however, can create a powerful record about transmission. Isolates of tuberculosis that are more closely genetically related are often considered to be more closely related in transmission. So molecular epidemiology studies that use fingerprinting or genotyping approaches may genotype isolates and then consider isolates with the same genotype clustered and potentially linked to recent transmission. Now, with increasingly available genomic data, we have even higher resolution, and we can use patterns of genetic distances, often numbers of SNP differences between isolates, to cluster isolates. Isolates within a given distance of each other are considered clustered and again potentially linked through transmission. So our inferences rely on the measurement of subtle differences between closely related genomes. And our inferences are only as good as our ability to measure these differences. Genomic epidemiology studies often sequence the bacteria directly from culture. And there are many sources of errors in these genomic epidemiology studies, from sample collection to variant filtering. And today I'm going to focus on our bioinformatic approaches. And these approaches vary among different groups with respect to many aspects of pipelines. However, whether different approaches identify consistent and accurate sets of variants is unknown. So here we investigated do variant identification methodological choices affect transmission inferences. And we looked at this two different ways. First, we reanalyzed previously published sequence data, and we asked, do different pipelines from different groups applied to the same data generate consistent inferences? Then, because we don't know the underlying truth of any true outbreak sequence data, we took a simulation-based approach to then investigate the accuracy of different variant identification approaches. So in our first part, we reanalyzed previously published data from a clonal outbreak in Germany. There were 86 samples that were sequenced in the original study. And the original study also, interestingly, Sanger's sequence confirmed 85 SNPs. 4 independent genomic epidemiology research groups took the same set of sequence data, applied their methods to it, and generated variant calls. We then compared these variants across the different groups' pipelines, and not only did we compare variants, but we also compared the transmission inferences made from those variant sets. Turning now to our results for this part, we found that different pipelines identified different variants in this same sequence data. So this is the magnitude of outbreak internal SNPs identified by each of the 4 pipelines in different colors A through D, compared to those identified by the original study in E. Not only did the magnitude of variation differ across pipelines, but so too did the identity of the variants. So again,
These are colored by the pipeline, and all groups identified a common set of 55 variants, but each group identified anywhere from 2 to over 200 unique SNPs. Sensitivity in recovering those original Sanger-confirmed SNPs also varied, and the maximum sensitivity was only 93%. These different variant calls resulted in different transmission inferences. And here is a violin plot showing pairwise distances identified by each of the pipelines. And at 5 and 12 SNPs are commonly used thresholds, genetic distance thresholds, to include or exclude the possibility of recent transmission. So if we apply those 5 and 12 SNP thresholds, we see that the different pipelines would infer different percentages of potential transmission pairs. So for instance, applying pipeline A would generate the conclusion that the majority of isolates are linked in recent transmission, whereas pipeline C, you would conclude that most isolates are unlinked. We then simulated an outbreak. This is the outbreak phylogeny, and then we simulated evolution over the course of an outbreak phylogeny. This generated a set of closely related Mycobacterium tuberculosis genomes for which we knew the underlying truth, the true patterns of difference between outbreak genomes. For each of these outbreak samples then, we simulated Illumina sequence data, generating the same kind of data we would generate in an outbreak investigation. For each set of sequences, for each sample, we then mapped those sequence reads with 3 different mapping tools, identified variants with 3 different variant calling tools, and filtered variants. So this generated, for each of these combination of tools, a set of candidate outbreak variants. And we could compare the candidate outbreak SNPs identified by each tool combination with our underlying truth to measure true positives, or true and false positives, and measure overall accuracy of our pipelines. Turning now to the results for this part, the simulations part, we see that performance, and this is false positives here, performance varies widely across the different tool combinations. Going across the x-axis are the different mapping algorithms and variant filters, and the colors indicate variant caller. Again, the dotted lines show commonly used thresholds of 5 and 12 SNPs. And the important thing is that even in tool combinations that apply filters, frequently the average false positive pairwise errors exceed these commonly used thresholds. So what this means is that the application of our pipeline alone, bioinformatic errors, can lead to the incorrect conclusion that there's no transmission. So clearly, some degree of variant filtering is needed because of this issue with false positives, but how much filtering should we do in our analysis? Filtering might exclude the false positives. At the same time, filtering will also reduce the true variation that we observe and our ability to infer phylogenies. To investigate the effect of filtering, we took a single candidate set of variants, and we applied increasingly strict filters based on quality score, such that each additional filter would exclude an additional 10% of variance. And as we would expect, as we apply increasingly strict filters, we see that false positive errors decrease dramatically. And as we would expect, as we apply these increasingly strict filters, false negative errors increase. And it seems as if with these particular parameters, there is some optimum level of filtering, and that depends, of course, on the weight of these different types of errors. We see a similar thing when we're looking at the accuracy of our inferred phylogeny. And again, as variants are increasingly filtered, at first filtering is important. After that, increased filtering rapidly erodes the quality of the inferred phylogeny. turning back to our original question, we asked, do our methodological choices matter? And we find in the reanalysis of this outbreak that different approaches can identify different variants. And this does result in different transmission inferences. In our simulations, we find that performance varies widely in different tool combinations, and that filtering is important, but does not always improve the reconstruction of the underlying true outbreak phylogeny. So ultimately, we are highlighting a problem for further discussion and suggesting that a standardized approach could improve inferences and our ability to draw comparisons across different studies. We also find that pathogen genomic epidemiology will benefit from pathogen-specific resources and tools. Thank you. This was the work of a group: Jason Andrews, Caroline Colleen, Ted Cohen, Julia Crota, and Barun Mathema, and I look forward to continuing the conversation during breaks.
Thank you, Catherine. Are there any questions? We have time for multiple questions. Megan?
Thank you. That's a very useful analysis. I'd like to mention the SNP thresholds, which have been sort of adopted by the TB community. So 5 or 6 SNPs seems to be possible transmission, and over 12 probably not. To me, there seems to be a danger that we might be optimizing our pipelines to fit those thresholds rather than the other way around. Maybe you would like to comment on that?
A great question, and I think there's been a lot of discussion on the use of these distance thresholds, and often they aren't designed for a particular study at hand, but they're adopted from other studies. And I think that there are other methods, clustering methods and phylogenetic methods, that are increasingly used, which I think are very promising. However, we need to think about what the optimal level of filtering is for those methods as well, as the phylogeny analysis showed. But I think that's a great question, and likely thresholds will differ across studies and in different contexts.
Over here, please.
So, Stefan Niemann, thank you very much for using our data for the fascinating analysis.
Thank you for the
fine.
data.
I have a question with relation to the error profile that comes from not so variable regions in the genome. So you would just need to set up like a core genome that is used, and then you would not have all these SNPs in the PPE, PGIS regions, and the different like filtering mapping characteristics. So is it basically just an effect of looking in the right part of the genome, or is there more beyond the variation?
That's a great question. There are several genes which are highly repetitive that you mentioned, the PPE genes, and we did look at the errors in those genes compared to the rest of the genome. And even excluding those genes from analysis, which is commonly done, does not entirely eliminate false positive errors, although the majority of false positive errors are found in those genes. However, if you exclude those genes, you also exclude a disproportionate amount of variation. And if our studies rely on very few SNP differences, we would like to harness all of the genetic variation in the genome. So ideally, our studies potentially using long-read sequencing or new variant calling algorithms could include these genes.
Megan? Okay. Any other questions? Thank you, Catherine.
I'm pleased to announce our next speaker, Dr. Megan Murray, who's a professor of global health and social medicine Associate Professor of Medicine in Department of Medicine, Harvard Medical School, and Professor of Epidemiology at Harvard School of Public Health. She directs the Department of Global Health and Social Medicine Research Corps. She is also Director of Research for Division of Global Health Equity at Brigham and Women's Hospital. Many hats.
Thank you, and I'm delighted to be here. Actually, this is a research project that was done in collaboration with Chuan-Chin Huang, who just presented. And I think for the sake of variety, you're just getting me this afternoon. So we're interested in the genetic variations of TB that are associated with TB transmission. So for the past couple of decades, I would say, my group has been interested in the various determinants of TB outcomes. So across the board, TB infection, TB disease, TB treatment, outcomes, and death. And we've looked at a variety of different contributions to that, including the host, so malnutrition, diabetes, smoking, etc., environment, health services, and most recently we've been focusing on pathogen. So what we wanted to do was ask the question, are there pathogen-specific determinants of TB infection, given that someone has been exposed to TB, and are there pathogen-specific determinants of disease progression, given that someone has been infected? So specifically, by transmission, we mean TB infection given an exposure. Of course, TB is transmitted as an aerosol. We measure TB infection either through a tuberculin skin test, as we did in this study, or through an IGRA. So we're asking the question, given that someone has been exposed, have they actually developed a positive TST or IGRA test? And then disease progression obviously assumes that someone has been infected and goes on to develop disease. Chuan-Chin introduced the study that we've been doing in Peru. This began in 2008. The goal of the study originally was to measure the transmissibility of drug-resistant TB. And in the process of designing that, we realized that there were many other things we could look at. We recruited index patients at about 106 different health clinics in Lima. And we went out to their households and recruited all the household members who would consent to be part of the study. At baseline, the index patients had sputum stored, they had a drug sensitivity test, and many of those isolates were later sequenced. The household contacts underwent tuberculin skin testing at baseline. They were examined for the presence of co-prevalent TB, co-prevalent with their index case, and then they were followed at 2, 6, and 12 months. People who were TST negative at baseline had a repeat TST at 6 months and 12 months to rule out infection. At the end of the day, we were able to calculate how many people in the household had been infected at baseline, how many became infected over the 12 months of follow-up, and how many developed disease during that period. So to do this study, to look at the pathogen-specific determinants of transmission and disease, we sorted this out into 2 different parameters. The transmission parameter is what we're defining as the proportion of people in the household who've been exposed to a person with active pulmonary TB that's culture positive and who become or are TST positive at baseline. And because we're concerned that people who are older may have been infected in the past and not due to the exposure in the household, we restricted this particular part of the study to people under 15 years of age. And then we also looked at a disease parameter, which we just said, is anybody in the household go on to develop disease, secondary cases of disease? And if they did, they were considered an event. If they did not, they were considered a non-event. So at the end of the day, we had 4,500 index cases. About 3,573 were culture positive. Many of those have now been sequenced. We looked at people, households that had contact under 15 years of age, and that narrowed the numbers down. And then we counted the numbers of transmission events and disease events that occurred. So there's another way to look at transmission, and it's been brought up a couple of times today, which is to look at molecular epi and to look at clusters as proxies of transmissibility. You can do this in a number of different ways, using tools like spoligotyping, MIRU, and whole genome sequencing. And actually, we did all of those in this study. We started out doing Spoligo and MIRU together, then went ahead with just MIRU alone when we found that Spoligo was not contributing any extra information. And now we've sequenced many of those strains, so we have both of those. And then, of course, we decide whether the patients are in a cluster defined either by similarity or identity of MIRUs. But in this case, we used an 8 SNP threshold to call people as belonging to a particular cluster. And then people who are unclustered did not share a whole genome sequence or even within 8 SNPs with any other of the strains in this large data set. So when we look at it that way, we found that there were 268 isolates there were 1,456 isolates in 258 clusters and about 853 non-clustered cases. And we chose one isolate from each of those clusters to include in this evaluation because we didn't want to over-represent the same genome multiple times. So once we have sequenced all these strains and we're asking the question, is there a SNP or is there a variant that's associated with infection and disease progression, there's a number of ways to do these analyses. In the past, we've used something called phylogenetic convergence to try to identify drug-resistant SNPs or novel drug resistance genes by genome-wide association studies. And that basically looks at whether SNPs have arisen in different parts of branches of a tree that have the same phenotype. So we have a bunch of strains, as you can see in this little circular graphic. If several strains in different branches have acquired the same mutation and have the same phenotype, we can infer that that's a gene that is associated with a particular phenotype. And there's a lot of statistical ways to test that assumption by simulating these data and coming up with a null hypothesis to test it against. More recently, we've been using another approach, which is a little bit more straightforward and grows out of human genetic epidemiology, which is to use to try to address the question of the relatedness of strains by coming up with a genetic relatedness matrix. So this is just a list of how much genetic variation is not shared by all the pairwise comparisons within the data set. And then one can include that as a term in a model, a straight-ahead linear regression model, a mixed model, to try to get at the population structure that we want to get rid of when we do this kind of analysis. So we don't want to say, oh, here's a particular cluster of strains that are spreading, and we think that because there's a SNP in that cluster and that they all have some particular phenotype, that the phenotype is due to that genotype, because it may be due to just population structure. So the goal of these analyses, which really kind of end up in the same place, is to get rid of that population structure. In the end, we don't really have to be as worried about very specific statistical cutoffs as we might be if we were doing human epidemiology, because we can take these strains once we've decided that we've have some putative genotypes associated with a particular phenotype, and we can make them in the lab and phenotype them compared to their wild-type predecessors. And really, that CRISPR and new methods make that quite tractable. So when we do that kind of analysis, we end up with a graph like this. And this is just a toy example to show you how this works. The top number A is a genome-wide association map. The x-axis is simply the genome, and the y-axis is the inverse p-value. So at every site, each gene or each locus across the whole genome, we get a particular p-value that indicates the association with the phenotype of interest. So in our cases, infection and the infection parameter and the disease parameter we mentioned. what we expect to happen is that we expect some false positives, right? We know that if you do 20 tests, one of them is going to be positive. If you do millions of tests, many of them are going to be positive. And we expect the distribution of p-values to range between 0 and 1 across the genome. So that first cumulative p-value distribution just says we expect that there will be a linear relationship between the number of mutations that we're going to see and a particular p-value. The QQ plot is, in the null hypothesis, the QQ plot will be linear, just like the previous graph. But if we see p-values that are actually lower, but looks like higher from the way this graph is, that are higher than that linear diagonal line, we can assume those are real hits. The value of the QQ plot is that if we see that there's a ton of real hits, we know that they're probably not real, that there are false positives that are coming up. So what we expect is that for any particular characteristic, we are going to see some, but not a lot, of true hits. So graphs should look something like this. Okay, so this is our data. This is just a QQ plot for one of our outcomes to show you what the effect of correcting for population structure does. You see in the top graph, we have a lot of positives, way more than you saw in that previous QQ plot. And we know that without adjusting for population structure, we're going to get a ton of false positives, and this data is really uninterpretable. But once we include a genetic relatedness matrix, we find that it falls into this nice pattern where we have a handful of mutations that have p-values that are consistent with a real effect. And so those are the mutations that we would go after if we were going to do experimental work. So here's some of our results. When we look at transmission as measured at baseline, we find that there's a number of mutations, a number of genes associated with that phenotype. Many of them are in the PE and PGRS regions. Some of them are well known as mutations, as genes that have been associated with interesting virulence factors, for example, ESXV. And one that was particularly interesting in this analysis was this ILVX. So it turns out that ILVX is quite strongly associated with transmission, but there weren't a lot of ILVX mutations in our data set. However, ILVX is part of a cluster of ILV genes that include ILVX, B1, G, and D. And when we looked, it took all of those genes and looked at that as a single phenotype, or a single genotype, we found it had a very good p-value of 10 to the minus 8th, suggesting that there is an association between this cluster and transmission. So this is now the disease phenotype, and we find that there's a lot of similarity. You can't actually get disease unless you're infected, so we did expect to see some overlap between the genes associated with infection and the genes associated with disease. And again, you'll see this ILVX in there in a small way, but across the cluster, we got a p-value of 10 to the minus 7th, so very strongly, again, associated with this disease phenotype. When we look at it this other way, looking at the more traditional molecular epi approach, the clustered versus unclustered, there is some overlap, again, between some of the genes that we saw. The ILVX cluster is still significant, but at a less significant level. So ILVX is actually interesting. It turns out that this cluster of genes are elements in the required for the synthesis of branched-chain amino acids, and to some extent branched-chain fatty acids. Branched-chain amino acids are thought in bacteria to be good metabolic indicators of the cell's general metabolic state. And we did find this one study that suggested that inactivation of one of these genes led to a branched-chain amino acid oxytrophy and attenuation of virulence in a mouse model. So that's a little disconcerting because we kind of expect to see increased virulence rather than decreased virulence. But this was a complete inactivation, and we don't really know what the mutations in our ILV cluster do. So we find this very interesting, and the next steps will be to try to make ILVX mutants or ILV cluster mutants and try to phenotype them to see if they have increased or reduced virulence either in a cell or animal model. So this is work that was done by a large group of people. Chuan-Chin Wang really was the leader of this analysis and would have been presenting if you hadn't already seen him already this afternoon. But many people contributed to our study in Lima, and you've heard about some of them already. The work was funded by the NIH, and we're very grateful to them and for being here. Thank you.
Any questions?
Okay.
Can somebody give the mic there?
Stefan.
Thank you very much. When I remember that correctly, then Dick van Solingen published a study last year on a similar question. So they also identified in the Netherlands genetic markers that are associated with transmission. So did you look into that study if you find similar markers?
I'm sorry, I missed the name of the study. Which study?
Von van Solingen, Dick van Solingen. I think it's a
Dick
paper.
van Solingen.
It's a paper in the Blue Journal. If
Yeah,
I remember that correctly.
we wrote that. We were co-authors on that paper.
But do you find similar?
We did find one of so there's one of the PGRS genes from that study, which I'll describe in a moment. There's an intergenic region that's implicated in this study. Is very close to that, one of the regions found in that study. That study was a very small study where we took, Dick van Solingen has a huge archive of strains from all the strains in the Netherlands that have been sequenced have been stored since they started collecting strains. And we were using a molecular epi approach, so we took strains that we thought shouldn't be clustered, but were clustered, and strains that we thought should be clustered and were not clustered, and took these extreme phenotypes, sequenced them, and then compared them. And we came up with 4 or 5 hits, even though there were only a couple hundred isolates in that study. And then we phenotyped those by looking at immunological responses in a cell model. So there was this one area of overlap, but I was going to present this as the round 1, and this is round 2, because there are inherent problems I think, with using the clustering approach to identify transmission. And the study was small, and we used phylogenetic convergence as our analytic tool, which both of these methods work, but they each have their strengths and their weaknesses.
Gandotra from IGIB. I was wondering if you've looked at the implication of these SNPs on the function of the gene. So is there a change in the expression level? Are these frame shift? What sort of mutations are these?
We don't know if there's a change in expression level. We're going to have to make the mutants, because that was the other piece to what we did with the earlier study. We pulled the strains from the archive and then phenotyped them. But what we'd really like to do is take a experimental strain, an RV or whatever experimental strain we choose, and then create a single point mutant that resembles the mutations that we found, and then compare them. So that will be part of the next step, and when we do that, hopefully we'll also do some transcriptomics, but we're not there yet.
Last question, please.
Okay, thank you very much. Very naive question. We have seen that there are several markers for transmission in PE regions, repetitive regions, but the current pipelines who, analyze the transmission based on SNP exclude these regions. Do you think it's time to move to, I don't know, longer reads to include also these regions because maybe there's a reason for variability there?
Yeah, probably. I mean, we certainly would love to be getting longer reads across the board for lots of reasons. It turns out that the PE genes have areas of variability and areas without variability, and so we trust the areas that tend not to be variable more than the other areas. But I think we're missing a lot. In many of our GWASs for different phenotypes, we find PE and PGRS in regions, and I think it's too easy just to dismiss them as hypervariable regions that don't have any meaning.
Thank you, Megan.
I would like to thank all the speakers for this afternoon session, for the great presentations, and for the audience for staying this late. We will now have another 15 minutes, I think we have left, for a panel discussion, and for that I will give the floor to Annelies Verrieg.
Please, Sebastian Gagneux, Ed Nardell, and Caroline Williams, come sit here.
Okay. Well, thanks for everybody who's still here, missing the vice president, but I hope you were rewarded by listening to interesting talks. It struck me when listening to this session that, especially when we teach, we try to totally simplify things. And I was reflecting on what I say when I teach, and it's smear 3+ is more infectious than smear negative, but there are studies where culture negative you still have transmission. And then that's one piece that I think we all teach. And then another piece is, you have these cavities, and as these cavities destroy an airway, and then when somebody coughs, a lot of junk from the cavity goes into the sputum, and that's also how you get a lot of transmission. And when you listen to this, you're like, whoa, this is so far off. And we're not only talking about larger airways and cavities, but we're talking about, interstitium, alveoli, larger, smaller airways, and it all has a different effect on transmission. And that's just the lung structure. Then we have, the lineages, the bugs, whether they're compensatory mutations or not. It's overwhelmingly, I think, becoming overwhelmingly complex and difficult to really piece it all together. But just as a kind of rounding up, especially with your study, Caroline, is it because with the breathing, it's more the alveoli and smaller airways, and with coughing, it's probably more the larger airways. So is there anything there? How can you relate that to the other things you've heard?
I think that's very true. I think as we're beginning to become more confident in the sampling of aerosol that's not sputum, and started using aerosol as the sample that could tell us more about transmission, we're certainly seeing more differences between what we see in aerosol and what we see in sputum. And I think, if I'm wholeheartedly honest, I think as with everything in life, nothing is black and white. And there's probably contributing factors from both small and large airways. And it may be that the bacilli that are excreted from the larger airways are excreted in sputum. But I think we have to remember that those are the bugs that aren't ever going to transmit to anybody else, because they're stuck in sort of extracellular gob that's just come out with it, and they're never going to manage to get into the air to transmit onwards. So the question has to then be asked, what makes the bugs that are in not in sputum that are in air different, and are they different? And I think we're beginning to see more differences, though I do appreciate that that is quite a big frame shift from the traditional teachings of sputum is indicative of what's in the aerosol, and I think we're going to see in the next few years much more information about those two things. I know Ed feels quite strongly equally, so it would probably be good to get his view on this.
Yeah, I'll be a little contrary. I don't think, I think what we've known for maybe 50 years about factors in transmission, that cough frequency, cough strength, viscosity of sputum, environmental factors, host factors, and source factors all come into the mix in terms of the how much transmission. I don't think any of that has really changed. I think still predominantly cough shears off aerosol and produces, which produces small particles, and that treatment and other things rapidly stop that. So I think what we don't know is some of the things we talked about is what are the adaptations to transmitting organisms, things like that. But I don't think that the basic, for example, Kevin Fennelly was mentioned, you mentioned him, and showing that cough aerosol correlated better with transmission than does sputum positivity alone. And that makes a lot of sense because it incorporates cough, which we don't normally incorporate when we just ask if somebody's smear positive or negative. You have to be able to generate an aerosol. So that really fits well. So I don't really think there's that much new in what we know about, there's been a lot of studies on cough now, and that still seems to correlate with transmission in my view. I think the interesting stuff on the organism we just heard is quite new, of course.
If I may, I think on a population level, that's still very true. I think epidemiologically, it's a numbers game, isn't it? Epidemiologically, over a large population, that cough and AFB smear grade, for instance, will correlate with transmission. But on an individual level, Eddie Jones-Lopez and Kevin Fennelly showed that actually the aerosol does predict transmission better, and we're seeing on an individual level many more cases of culture negative TB that transmit, patients that don't cough that transmit, patients that don't produce sputum that transmit. And I think those are that's where we're seeing this grey matter between the black and the white, and I think it's probably a little bit of both. I think the idea that you have to cough in order to produce particles and aerosols within the respiratory range that cause transmission is beginning to be debated. The Worry paper that looked at, admittedly, at TB patients with just tidal breathing showed that they had a 3.5-fold increase in the amount of particles within the respiratory range that were excreted. Now, she didn't go on to check that for transmission, and that's the next big question, isn't it? And that's where the next data should be going. But the evidence is pushing that door that maybe there is a difference on an individual level.
Yeah. So moving, okay, you want to say?
I'm just, I'm really not the expert of aerosols and coughing, etc. But usually I claim, also teaching, that TB has to cause disease to transmit. And I wonder whether this is still true. I mean, if you compare to other pathogens, this is obviously not the case, right? I mean, there's many other bacteria, for instance, that you carry in the nasopharynx, which just transmitted all the time, totally independent of virulence, in other words, of disease. And I always thought that TB was interesting because there is actually a link between disease and transmission. So there's all kind of downstream consequences of this famous trade-off between virulence and transmission, and you have kind of the balance. Don't be too virulent because then you kill your host too quickly so you don't transmit, etc., etc. So I was wondering what you guys thought about this. I mean, does this mean that this is not true anymore? So if we don't need the cough to transmit, or I mean, we have also these prevalent cases in the prevalence survey where we find people who are smear positive but don't have any symptoms. How does that relate to disease, or it's just very early disease?
So one thing we haven't talked about is the probabilistic aspect of transmission. And the TB organism has evolved to get from one person to another by a very efficient mechanism in terms of being in air and not requiring close contact, but at the price of dilution. In Riley's studies, it was one particle in 30-some cubic meters on average. And that requires an awful lot. I mean, on average, it took a student nurse about a year to inhale an infectious dose. So, because of incredible dilution. So, that would suggest that you need to have an awful lot of organisms to be infectious. Now, the exceptions occur. Smear negative, very little disease, environmental differences, strain differences, all can account for that, for the exceptions. But the general rule, I think, is you need a lot of organisms, cavitation, coughing, to produce the numbers of organisms that increase the probability of transmission.
I think in certain populations where we contact trace everybody, if it required disease, if it was only that you had to have disease in order to transmit, would we not have eradicated it in those populations where we contact trace absolutely everybody to prevent onward transmission? So there must be a subset of people who don't have disease overtly, and these are our preclinical patients or our patients with sort of transient disease, who feel well, don't report to hospital with symptoms, but are probably likely transmitting forward. And we're not at the moment diagnosing those people early enough. They are missing millions, if you like, but maybe they don't have disease. Maybe for them it's a transient state that their immune system will deal with and they will get better. But during that period of time, they would onwardly transmit. So I don't think you do need overt disease to transmit, personally.
Before we move to something else, is there somebody in the audience that has an opinion on what is the contribution of incipient or subclinical disease to transmission? There you go.
I think in the end, the epidemiological question is contribution to transmission. So you can have, you will have clinical phenotypes probably who have very low production of infectious particles and who will, for example, if you look at it in the study designs that we've heard today, which are household contact studies, you will see a low number of secondary infections. But at a population level, it may be quite different. If you are in a situation in everybody who coughs, or the majority who coughs, are actually diagnosed and put on treatment. So then those who don't cough can be overrepresented in your population, which is what we're already seeing from the prevalence surveys. So even if they have a lower rate of transmission, they may in the end start contributing to a relatively large extent to transmission. And I think that is the situation that we are starting to face now. And I was actually intrigued by the data that came out recently in the New England of this large population study in Vietnam by Guy Marks, where he actually showed that he could really reduce over a period of 3 years by screening, not just screening, doing gene export on everyone in the population for a period of 3 years annually. He could really reduce not only prevalence, but likely also transmission, whereas other studies haven't shown that. And I think the difference is, or the potential difference is, that the other studies actually only did symptom screening, so they didn't screen everyone. So this, I mean, it's not direct evidence, but it suggests that indeed the ones that would otherwise not be screened out because they don't have symptoms actually contribute to transmission, and you actually have to get them in order to really reduce transmission from a high to a lower transmission setting, if with all other things remaining equal.
Okay.
But does anybody know of actually evidence, direct evidence, like in the classical, let's say, Molepi way of things? We say, okay, yes, this person had, asymptomatic TB and infected this person who now has symptomatic TB. Is there anything known along those lines? Marcel knows.
Marcel, yeah.
Yeah, so my answer is not as sophisticated as Frank. This is more anecdotal. But in our JID paper in the Inuit community, some of the people were identified as contacts and they were asymptomatic. And then a TST was done and was positive, so then an X-ray was done and there was a cavity. And one of those who had 3+ AFB positive, who was asymptomatic, could then be inferred by genomic epi to have started the cluster of 13 people. So this is just one anecdote, but I think this is quite plausibly going on in many parts of the world where we start off screening for symptoms, but this person was just picked up as a contact and not as a potential source until the genomic epi worked it out backwards.
But not having symptoms and not having disease are two different things.
Exactly.
So I think missed disease is a big factor, yeah.
Exactly.
Okay. All right. I want to move to the bug now. Sebastian, you gave a very nice talk on compensatory mutations. So you have a drug-resistant mutation, it causes a hit to the fitness and potentially infectiousness, transmissibility of that bug, and then you get compensatory mutations and you get back to pan-susceptible. So that's a nice model to study, but are there, so we've seen many other examples now of changes, genomic changes that can be related to infectiousness, but are there equivalents in drug-susceptible TB where we see a hit due to some mutation, something that happens to the bug, and then we get some compensation to overcome that? Or have we only seen that in drug resistance?
So you mean that some mutations happen which are bad for the bacteria and then you need some kind of compensation?
Yeah, related to transmissibility.
Yes, so I mean, just generally, most, as you might know, most of the mutations usually are either neutral, so they don't have an effect, or they'll be slightly deleterious, generally. And so presumably, there's always mutations that can occur that are actually slightly deleterious for whatever phenotype you're going to measure. So presumably, if they're extremely deleterious, the bacteria would just disappear. If they're only slightly deleterious, you can imagine that you need to somehow invent a compensatory mutation to actually compensate for this slightly deleterious mutation. So I would assume that some of the mutations that we discover when we sequence pan-susceptible strains would be slightly deleterious mutations, which just have happened, and there's nothing that the bug can do about it. And then there's some perhaps other mutations which compensate for this, but actually to figure out which one is which, I mean, that's extremely difficult because we don't really have the phenotype. Now you say transmission phenotypes, Megan just showed us an example, right, that presumably there are mutations there which somehow increase or maybe also decrease transmissibility. The question is, what is adaptive, right? I just say the bad mutations just happen, so that presumably it's not adaptive, it's just genetic drift because you know, bad things happen. Still, you need to perhaps compensate for that. But then there might be selection for hyper transmissibility, and I think that's much more difficult to, in the context of drug-susceptible strains. Just generally, if you have a certain, let's call it fitness, transmission fitness, and what is the driving force for a bacteria to acquire even more increased transmission? I think that's difficult to find out, but yes,
but I guess we would have to look for pairs of mutations that frequently occur.
It could be much more complicated than just pairs. I mean, if we take average 2 strains, right, they can have like hundreds of mutations, and these mutations would interact in all kinds of different ways. I think that's going to be extremely difficult.
Yeah. Megan?
So there's very little that's a selection pressure like drugs, right? When we look at the p-values for when we do a genome-wide association study on on a set of whole genomes, and we're doing it against drug resistance, we get astronomical p-values of 10 to the minus 20th or whatever. But no other, and we still get significant p-values for other kinds of phenotypes, but nothing like drug resistance. So I feel like there's there's not much that TB encounters in its daily life that puts the kind of selection pressure on it that drugs do.
Okay. I think we're rounding up time, so I just wanted to see, so we have 5 more minutes, so I wanted to invite some questions from the audience to the panel.
Just Philip Hill. This is a question for Sebastian. You may have mentioned it in your talk, but I'm a little confused as to how to set criteria for compensate that will define a compensatory mutation.
Can you repeat? Can you repeat? I didn't, just louder.
Louder, okay. So the question is how to set a criteria that will define a mutation as compensatory.
Yes, okay, that's a good question. There's multiple ways you can do that. The complicated way is the one that, again, Megan mentioned, that you actually take these mutations, put them into the H37RV and actually prove genetically that these mutations do what you think they're doing. Obviously, we haven't done that, particularly not because we're facing at least 90 different potential compensatory mutations. Now, some people have done that with other bacteria, as I mentioned, these same or very similar mutations you can actually see in other bacterial species, which are much easier to manipulate genetically. This was actually done and proven to actually really have this effect. Now, in vitro, of course, you have a resistance mutation, you add the compensatory mutations, then the strains grow better. It was done in Mycobacterium smegmatis, but just for a few number of these mutations. We used, at the time, also an indirect, which along the lines of, again, Megan suggested, this conversion evolution idea that because TB has very little diversity generally, and it's totally clonal, so you wouldn't expect mutations to occur multiple times independently unless they do something. Not just do something, let's do something important, so that relates to the pressure, so drug pressure, or the extremely strong fitness pressure. So essentially, many, not all, many of these mutations that we see have a very strong signal of convergent evolution. So they keep popping up again and again in different unrelated strains. So that's a very strong indication that they're very strongly positively selected. But that's about what we can do. Then we can learn, of course, in terms of the structure, where they actually occur in the RNA polymerase. It's near the RNA exit channel when you say, okay, that's, yes, very likely to actually have an effect based on what we know from smegmatis and other bacteria. Then we can have different classifications of these compensatory mutations and how confident we are about the effect of those. And so we're just starting to do that. And in fact, if you actually take the mutations which we are most confident about, the effect actually becomes even stronger. But currently, it's just like we say putative compensatory mutations.
Okay, thank you. And I think it's time to round up. I want to thank everyone for attendance and all the speakers of the panel, but also all the other speakers for an excellent session this afternoon. Thank you.
You can go first. Okay.
I could go first.
Okay. It's very
So before you leave, this brings TBScience 2019 to an end. I think it's been a wonderful meeting, although I already noticed that the last few hours were a bit less crowded than in the beginning. I think we have had some difficulty with competing sessions, so we'll try and work on that. For those who don't know the history, we just started with this last year, and this was last year meant as a pilot. and it was a tremendous success. And we thought, okay, we can do this once, let's see if we can do this twice, and I think we managed to do this, so I think it's great. We've had wonderful sessions, we've had wonderful speakers, we heard a lot of news, and I think what this meeting is really doing is what it was meant to do, is give a broad overview of what's happening in science to everyone who's interested from the TB community, but also to allow TB researchers from various walks of disciplinary life to get in discussion with one another about sort of the bigger problems in tuberculosis, the natural history problems and all the issues that we see. So I think this has, for me at least, really worked. I think we've been very successful in that we had over 700 registrations. And we had a very large number of abstracts. We had, as you've seen, a large number of great oral presentations, and we had wonderful poster presentations. I wanted to thank the union for basically taking the risk, because when we started discussing this 2 years ago, I think the question was really, are we willing to take this on? And the union did, and I think that's really great. And they've shown a lot of commitments to this conference. I wanted to mention specifically Paula Fujiwara at the union, who has been the driver behind this, but also Grania Brigden, very important in this respect. And we couldn't have done it without Joanna in the back there, who everybody who is a speaker here knows Joanna, because she's been chasing you and done a wonderful job. I personally hope that the union will continue this. We will look at a format that will work for everyone. And I would say, I mean, advocate for this, guys. I mean, if you like this meeting, advocate for it. Tell the union this is great. We should continue whichever way we want to. And now the floor is yours.
Thank you, thank you. I won't keep you long, but I think I just want to add to the multiple thank yous. This wouldn't have been possible without the work of the scientific committee headed up by Frank, Mark and Manjula. You'll also see the full list on the front page of the programme. I think if you have an opportunity to see them around the conference, to say thank you for the immense amount of work that they put together to put this really interesting and flowing programme. So this was not possible without them, so I'd like to thank the whole scientific committee, as well as the organising group. Joanna was part of that, and there's a number of people who've been helping with the logistical part of the meeting, so I'd like to thank them. And the volunteers who I think have left, but if we can pass on our thanks to them for sort of helping us with the logistics of the day. Then the other key sponsors who are really, who have supported the TBScience and have made it possible for a lot of people to be here is our sponsors. So the Bill and Melinda Gates Foundation, EDCDP and ATS have all supported TBScience, and so we'd like to thank them for that. And then, as we say, this is year 2, and I think we are being bold enough already to get a date for year 3. So put it in your calendars, tell your friends. Seville, 20 and 21st of October is the place to be for TBScience 2020. So we look forward to seeing you all again. There will be a survey sent out. This is a young conference, so we are very open to feedback. We want to make sure that we give you the conference that you would like. So please do fill that in, and we will try and take on board all of your thoughts. So thank you very much. And now there should be transport waiting outside to take you to the main conference opening. And as I said, after that's done, there will be drinks. So thank you.